Hello, everyone, and welcome to Advanced 2022 Sanfilippo Community Conference. I hope you're all doing well, and we're so glad that you could join us. I'm Glenn O'Neill, president of Cure Sanfilippo Foundation, and also husband to Cara, father to my daughter Eliza, who's age 12, with Sanfilippo type A, and my healthy son Beckham, who's age 15. Eliza was diagnosed in mid-2013 with Sanfilippo, and a few months later, my wife and I founded Cure Sanfilippo Foundation to move Sanfilippo syndrome research forward. A lot of progress has been made thanks to many foundations, several who came many years before we even started. Progress has been made over the years thanks to the scientists, the clinicians, researchers, biotechs, therapists, families, parents, and many more. These groups are all represented here over the next couple of days. A few years ago, our foundation began looking into having a conference like this to bring together these various groups in the Sanfilippo space. We felt it was really important to gather together to discuss the disease Sanfilippo specifically. And as we began having these initial discussions about doing this, the pandemic happened, and as we all know, the world shut down. But here we are now, all a couple of years later, and we're glad to be able to have this conference and put it together now in a virtual setting, which is allowing many more people to participate. Over 350 are registered for this conference, so thank you all so much. We have more than 50 speakers and panelists. And as we shared in our registration messaging, kind of the purpose of this conference is parents and caregivers of children of Sanfilippo don't often get to see the many brilliant people working behind the scenes in the efforts to make a difference for their children. This is the scientists, the researchers, and many more, as we discussed. Additionally, many working in the field seldom have the opportunity to spend time directly with the families impacted by Sanfilippo, the parents especially, to better understand and hear in detail and from multiple parents, what the daily lives are like, and to be able to use this knowledge to enhance their work. We look forward to high-level collaboration over the next couple of days between the speakers, the panelists, and all of you participating. There'll be information sharing and Sanfilippo-specific learning sessions. Topics will include the role of inflammation and the immune system in Sanfilippo, targeting heparan sulfate specifically, new treatment approaches, Sanfilippo subtype-specific sessions, clinical trial updates, ABA and Sanfilippo, parenting siblings of children with Sanfilippo, and much, much more. I want to give a big shout-out thank you to our Advanced 2022 sponsors and partners in the Sanfilippo space. So thank you to Denali Therapeutics, Lysogene, Alivex, Ultragenics, and JCR Pharmaceuticals. We are thrilled that you are interested in helping better the lives of children with Sanfilippo syndrome, and thank you for your work. We have a mutual goal of getting helpful and safe therapies to children with Sanfilippo syndrome as soon as possible. I want to take a minute just to thank some other folks. I want to thank our internal team here at Cure Sanfilippo, specifically behind the scenes, Katie, Lindsay, and my wife, Cara, and all the work they've put in to make this conference happen. I'd like to thank Kim Stevens and the folks at Project Alive for giving us some information and some great tips and some lessons learned on how to put together a virtual conference. It's all actually been very, very helpful. Another big thank you to the presenters and the panelists you'll be hearing from over the next 2 days. To dedicate your time, your talent, your insight, and wisdom to these important topics is so gracious of you, particularly with everyone's crazy schedules and most of you during the summertime break. I want to thank all the attendees who we're seeing in the comments section. So please, we'd love to hear from you and where you're from. Thank you for joining us today. We believe progress happens quicker when we have more people, more awareness, and more pushing this research forward. So thank you for your interest. Before I get started, I want to turn it over to my number one collaborator, my brilliant wife, Dr. Cara O'Neill, a pediatrician, and also our Chief Science Officer. Cara?
Thank you, Glenn. And for those who know us personally, you know that I couldn't ask for a better partner in everything that Sanfilippo has sent our way. And I'm so thankful for everything that you do on a daily basis and over the last 9 years to propel this foundation forward. The work of Cure Sanfilippo Foundation began 9 years ago, as you said, with the diagnosis of our daughter, Eliza. And over those 9 years, we have seen a tremendous increase in research activity into Sanfilippo. We have seen programs move from basic science to advanced clinical trials. This ramping up of activity, awareness, and funding has happened because of people like you who are here today with us, and so many more that support our mission. There has been a swell of energy and voices raising up to say that we must take action, and urgently. We have to think about this disease in new ways. Today and tomorrow, you will hear from experts about some of the fascinating breakthroughs that have come about because they reconsidered traditional dogma. They tried something new and discovered insights that are opening up new treatment strategies. And while we've made progress, we must also recognize that in the same time frame, many clinical trials have ended and companies have abandoned their work in Sanfilippo. It's important to make the data available from those studies to the scientific community so we can dig in and really learn from that and take those learnings forward. We owe that to the families who participated. It's critical that we challenge the paradigm of how we assess benefit and how we consider the use of biomarkers in clinical trials so that we can be successful in developing a system of treatments that can meet the needs of all children, regardless of their age and the level of their disease progression. Collaboration and data sharing will be essential, and an increased flexibility in academic thinking and regulatory action will be necessary to meet this challenge. In our collective mission, each of us has specific areas of expertise, roles to play, and contributions to make. We are so appreciative of each of you here for your contributions and your commitment to our community. And for those of you who are new to our community and maybe just finding your footing, please know that you have a place here, that you are needed, and you have the ability to create a better future for the children, all of us together. Over the course of this conference, we hope that the presentations you hear will stimulate new research questions, broaden your perspectives, provide useful information, and encourage collaboration. And all of this to advance Sanfilippo. Back to Glenn for some housekeeping.
Thank you. Yes, we do have some housekeeping items before we get started in just a few minutes. So the first is we have a packed agenda, so we're going to do our very best to keep to the schedule and respect the presenters and panelists' times that they've graciously blocked for us. And while we may be involved in some great discussions at that time, we are going to have to cut off at our intended ending times to stay on schedule. We know you understand. Second, you'll all see on your screen, there's an area to the right for a chat and a Q&A. As we will be taking questions during the sessions, please type in your questions or comments there. We'll be monitoring that feed and trying to ask as many questions possible as we can, time permitting. If we aren't able to get to your question, please reach out to the presenter directly or reach out to us and we can help direct you. And please do use this opportunity to ask your questions while we have access to these valuable and expert speakers. If you're thinking of a question, you know some other folks are probably thinking of the same question. And so please do ask so we can delve deeper into some of these important topics. And a quick reminder, while you're asking those questions, please let us know who those questions are directed towards. That would be a really big help. Third, there's a speed networking feature on the left side of your screen under People. During breaks, you can link up with anyone in the conference for a few minutes. So remember that. And also remember on the left side of your screen, you can visit the expo hall at leisure during your breaks. You can access this on the left side of your screen. Just click Expo, and you'll see some more about the groups who are presenting over the next couple of days. And there'll be a half hour set aside tomorrow to be able to spend some time specifically at the Expo Hall. Okay, we're going to start in just about 2 minutes to kick off at 9:15. So grab a quick coffee. We're getting ready to go in just about 2 minutes. Thank you again for joining.
Thank you. Hello, everyone. I'm so excited to kick off with our first session of this conference, Research Happening Now, Targeting Heparan Sulfate. And we are grateful to have with us today Dr. Luigi Pavone, Professor of Biochemistry, Molecular Medicine, and Biotechnology at the University of Naples, Federico II in Italy. Immediately following will be Dr. Karl, CEO of ReMotorTherapeutics. And concluding this session's presentations will be an amazing team from the Telethon Institute of Genetics and Medicine in Naples, Italy, Dr. Diego Medina, Dr. Antonella Capuozzo, and Dr. Giancarlo Parenti. Dr. Pavone, the stage is yours.
And so good morning to everyone. It is a great pleasure for me to be here today, and I would like to thank Cara and Glenn and all the Sanfilippo Foundation for having organized this wonderful meeting, bringing together families, scientists, and companies, talking about novel mechanism and novel therapeutic approach for the Sanfilippo disease. So next slide. So far, we have been working at the development of a novel therapeutic approach for the Sanfilippo disease based on a novel target, which is the extracellular accumulated heparan sulfate. With this therapeutic approach and with the use of a recombinant protein that masks the excess of extracellular accumulated heparan sulfate, we aim to reactivate the cellular signaling in order to cure the lysosomal disease, which are shown here. Next slide, please. But in order to understand this innovative therapeutic approach, different from the previous one, we need to little bit review the pathogenesis of the disease. The mucopolysaccharidosis are inherited metabolic disease due to the accumulation of undegraded GLUGs within the lysosome and on the cell membrane. They are autosomal recessive disorder, except for the MPS II, which is X-linked, and the clinical feature includes developmental delay, multiple dysostosis, organomegaly, a neurological disorder, and cardiac defects. And as you can see in this classification, they are classified in 7 subtypes if we consider the accumulated GAG, and 11 subtypes if we consider the missing enzyme. Let's go to the next slide. But what is most interesting is that the phenotype of each MPS subtype is strictly related to the accumulated GAG. So if we think of the GAG just trapped within the lysosome, we cannot explain this difference in the phenotype of the different type of the MPS. Instead, if we think at the biochemistry of the GAGs, of the GAGs and the proteoglycan, we can explain the phenotype related to each type of the mucopolysaccharidosis, and especially of the Sanfilippo disease, which has neurological involvement, where the accumulated substrate is the heparan sulfate. So let's go through. So GAGs are long-chain sugars made by the repetition of the saccharide unit, but they are mainly attached to a core protein, which is attached to the plasma membrane, being integral through an integral protein or a GPI anchor, or they are the constituent of the extracellular matrix. Let's go through. Next slide. And especially the heparan sulfate regulates many cellular processes, including cell proliferation, adhesion, migration, and angiogenesis. But how biochemically these GAGs regulate this process? As you can see in this picture on the left, the heparan sulfate mediate the interaction of the growth factor with their connected receptor in order to stimulate the activation of signaling cascades relevant for the brain development, synapses formation, and so on. And as you can see on the right, you can see that heparan sulfate also is important, being a reservoir of growth factor. So next slide. As you can see in this picture, Heparan sulfate, especially with the signaling of the fibroblast growth factor of the growth factor receptor, function as a bridge molecule between the morphogen in pink and its receptor. Without the heparan sulfate, the FGF cannot bind to its receptor and cannot activate the intracellular signaling, as is also shown in the crystal structure of these three compounds. And this signal is already active at the pre-implantation stage, where during the embryo morphogenesis, since the beginning, the HS, the heparan sulfate, and the FGF signal are needed for the initial phases of the embryogenesis. So next slide. But mainly what I want to highlight is that the heparan sulfate, proteoglycan, are important for the neurogenesis, interacting with different receptors such as FGF, EGFR. They are important in axon elongation and synapses formation. So what does it mean? If the levels of the extracellular accumulated heparan sulfate are deregulated, such as in the Sanfilippo disease, the neurogenesis from the first phases of the postnatal development is already impaired during the growth of the babies, bringing to neurological defects. So if we target the accumulated heparan sulfate, we might restore the neurogenesis during the postnatal development. Next slide. But what I want to point out is that even a mutation in the genes of the synthesis of the heparan sulfates, also in this case, you can have a brain development. What does this mean? That even if you have a lot of accumulated heparan sulfate, or even if you have less heparan sulfate, in both cases you have brain development. This means that the pathogenic mechanisms are regulated by the level of the heparan sulfate prior of their accumulation within the lysosome. So next slide. So our hypothesis starts from this scheme that was reported in Nature Review Molecular Cell Biology, where they say that if you have a proper balance between morphogen, GAGs, and the receptor, you have a proper activation of the signaling for the postnatal development and the normal growth. If you do not have enough GAGs, or you do not have synthesized the core protein, the morphogen cannot bind to their receptor. So you have inactive receptor and then develop pathology. But in the case of the Sanfilippo disease, where you have an accumulated, extracellular accumulated heparan sulfate, you have that morphogen that are trapped by the excess of the heparan sulfate and cannot bind their ligands, their receptor. So the intracellular signals are inactive, and so you develop the Sanfilippo pathology. Next slide, please. This was reported also by our colleague and president of the ASGLD, Brian Beaker, who showed already that even the hematopoietic stem cell transplantation didn't work in mice with MPS I through the impairment of the chemokine CXCL12, because it is trapped by the excess of accumulated heparan sulfate. It doesn't allow the homing of the stem cell transplanted to the right place into the body in order to cure the disease. So this accumulated heparan sulfate is pathogenic even in other therapeutic approach. So next slide. The idea that focuses on this accumulation of the heparan sulfate is that when you have a deficiency in the lysosomal enzyme, you have an enlargement of the lysosome, which lysosome, instead to be segregated, goes towards the nucleus in the perinuclear region of the cells, and there gets accumulated even more. And this accumulation block the endocytosis of the membrane molecule, and so brings to the accumulation of the heparan sulfate on the plasma membrane. So next slide. So we have shown on fibroblasts from MPS 1 and Sanfilippo B patient that you have an accumulation of the heparan sulfate on the cell membrane of these fibroblasts. So we start to look at the mucopolysaccharidosis more than a lysosomal disease, and as diseases of extracellular accumulated GAGs. Let's go next slide. Our therapy and our therapeutic approach is based on the use of a recombinant protein that masks the excess of accumulated heparan sulfate proteoglycan on the cell membrane and allow to reestablish the physiological equilibrium between morphogen, receptor, and heparan sulfate proteoglycan in order to have a normal growth and in order to recover from the Sanfilippo disease. Next slide. To this aim, we have used a recombinant protein, which is the NK1 protein that we express and produce in Pichia pastoris expression system. And here down there, you can see the crystal structure of this recombinant protein with the heparan sulfate proteoglycan. We have chosen this NK1, which is the NK portion of the hepatocyte growth factor, which has been discovered as the first ligand of the heparan sulfate, or better, the heparan sulfate was discovered as the co-ligand of the hepatocyte growth factor, which with its receptor. So that's why we choose this molecule that highly bind the heparan sulfate proteoglycan, so our therapeutic target, that we purify with an heparin column, due to its high affinity with the heparan sulfate. So now let's go through our results. So how does this protein work on cell system MPS fibroblast and Naglus stable clone, and in the Naglus, so MPS 3B Sanfilippo B knockout mice? And let's see our results in the next slide. Next slide, please. So first, we started with measuring the therapeutic efficacy of our NK1 molecule in MPS Sanfilippo B fibroblasts. And we did that at the very beginning of our research, the experiment of the incorporation of H3 glucosamine in order to measure GAG accumulation upon treatment with NK1. And as you can see, at a very low dose of 1 micromolar, you have a reduction of about the 50% of the accumulated GAG into the Sanfilippo B fibroblasts after 48 hours. Then we tested this therapeutic approach also on other MPS that were accumulating heparan sulfate and dermatan sulfate, because this protein has a high affinity, the same affinity also for dermatan sulfate, but not for chondroitin and keratan sulfate. And as you can see, this molecule worked on the MPS accumulating heparan sulfate and dermatan sulfate, but not in the MPS 4, which accumulates other GAGs. And here are just other tests with another method, like the Alchan Blue method, and the last panel shows that another compound, like the fibronectin, was inactive in restoring in reducing GAG content. So it is specific the action of our molecule in reducing GAG content. Next slide, please. In this picture is shown that not only we are able to reduce GAG as measured in that way, but we are able to reduce also the sites of the lysosome. As you can see in this picture, MPS 1 and MPS 3B Sanfilippo B, when treated with the NK1, show a very small and reduced lysosome as compared to the enlarged one in the MPS 1 and 3B fibroblasts, patient fibroblasts. So next slide. Moreover, we are showing the same experiment than before, but here we are showing that we have a reduction of the heparan sulfate staining on the plasma membrane of the fibroblasts of patients affected by MPS 1, 3A, and 3B. And together with these, after NK1 treatment, you also have a normal distribution of the lysosome all over the cytoplasm, where they are not more concentrated into the perinuclear region of the cell, but they are all distributed all over the cytoplasm of the cells. So we have reduced the appearance of fate, but we have also reestablished the normal trafficking of the lysosome in the diseased fibroblast. So next slide, please. And this was the experiment that confirmed our mechanism of action of this therapeutic compound. As you can see in lane 2, both in MPS 1 and MPS 3B fibroblasts, the FGF is inactive when given to MPS 1 and MPS 3 fibroblasts. But if you consider the same amount of the FGF2 together with NK1 in lane 5, you already have a signal. And this signal does not increase because giving that amount of NK1, you have balance the level of heparan sulfate. And this is the heparan sulfate that activate that receptor and that signal. So next slide, please. So those are the conclusions. Let's see later. Let's move to the in vivo experiment. So we have treated the Sanfilippo B mouse model every week with 10 milligram kilo of our protein with the intraperitoneal injection in this animal, and we have evaluated urinary GAG content, movement, organ size, histopathology, and neuropathology. Let's see the results. So as you can see, GAG content in the urine after 2 months treatment was reduced up to 38%. And as well, we had a reduction in the liver content of GAG as measured by Alsham Blue in this slide. Next slide, please. So here is a short movie that show that a treated animal was on the top, was exploring all over the cage. Instead, the untreated was stuck in the same place. Okay, I think this is enough. We can go to the next slide. And the next slide contains graphic images with organ. And this image was very clear to us because mice with Sanfilippo B has a very strong urinary retention. And as you can see, upon NK1 treatment, the urine was completely not anymore accumulated into the urinary bladder and was reduced. And this was for us a sign that the therapy was really working. Next slide, please. This is organ histopathology of the liver and kidney. Next slide, please. Here, I want to focus on this. I want to show you that about the neuropathology, that we have reduction of the theoflavin staining into the brain, to the cerebral cortex and the striatum of the MPS 3B mice treated for 6 months. Next slide. We had a reduction of the neuroinflammation with the GFAP staining and reduction of LC3. Next slide, please. But we have also reduction of heparan sulfate accumulation. As you can see, the treated KO mice, NAGLO-KO mice, show strong reduction of the heparan sulfate into the cerebral cortex. And the next slide, also of LAMP1. Next slide. Those are the conclusions. It's okay. Then I want to show the last slide about that show more insight into the mechanism of action. We have generated a neuronal model by silencing the NAGLO gene of the Sanfilippo B into the neuroblastoma cell line, and we have reproduced the lysosomal defects in this as you can see by LAMP staining. Next slide. The Sanfilippo B neuroblastoma cell line were unable to differentiate under retinoic acid treatment. But after addition of NK1, the cells differentiate as the control cells. But more interesting is that the lysosome are dispersed all over the axons because their fundamental is to distribute molecule and signal all over the axons. Next slide. We have shown also that the accumulation of anti-amyloid beta 142 peptide is strongly reduced in these cells after NK1 treatment. And the last slide, that lysosomal secretion after 36-hour treatment with NK1 is completely restored in these cells. So let's go to these slides. So we have shown that our therapeutic mechanism of action is that we have a compound, a protein that binds specifically a novel target for this disease, restore the FGF signaling and other morphogen signaling, reduce the Heparan sulfate proteoglycan, stimulate lysosomal isocytosis, and GAG and lysosomal reduction in this disease. And those are the advantages of these molecules, that is just self-produced by our body, so it is not immunogenic, it is able to cross the blood-brain barrier and to cure the neurological defects, and it is based on low cost of production in a fermentative way, and is working at a very low dosage. And so finally, I want to thank the Sanfilippo Foundation and my colleague and friends, Professor Pignataro, working at the neuroscience department, and my research group in the middle, and Professor Fraldi from Change, and Antonella Monaco, and Telethon Biobank for giving us patient fibroblasts. And I will be happy to take any question from you. Thank you.
I would like to thank Dr. O'Neill and the organizers for the opportunity to present this work today. As you all know, enzyme replacement for lysosomal storage diseases works. But for neuropathic ones, the problem has been insufficient delivery of the enzyme into the brain. To overcome this limitation, we propose to directly transplant into the patient's brain human neural stem cells that continuously pump out the target enzyme for the life of the patient. In this project, we aim to develop this cell therapy for MPS IIIA, missing SGSH, N-sulfur hydrolase, or aka sulfamidase. This approach is sometimes called ex vivo gene therapy, since the neural stem cells are genetically engineered by using viral vectors to overexpress the target enzyme. Creation of this therapy takes 5 main steps. First, we genetically engineer a stable neural stem cells using recombinant lentivirus. The lentivirus integrates into the cell's chromosome so that the overexpressing cassette is permanently placed in each cell and is not diluted away or lost during many cell cycles. This step is an overnight step. The next day, we wash off the virus and start expanding the lentivirus exposed cells. We create several tiers of cell banks that are frozen down along the way, the last tier being the clinical lot to be infused into the patient's brain parenchyma. Once transplanted, the cells will migrate throughout the CNS and continuously secrete the enzyme for life. This cell line, once proven effective, will serve millions of patients. It takes about 6 months to make the cell line, another 6 months to test and characterize it. Shown in the middle panel is MR images of a chronic stroke patient whom we treated in a phase 1 clinical trial by directly injecting neural stem cells into the stroke cavity. As shown in the baseline, this stroke cavity is clearly visible. 6 months after the stem cell injection, you see emergence of a new tissue inside the cavity. At 12 months, this new tissue has enlarged, and at 20 months is almost filling the cavity. This patient was immunosuppressed only for the first 28 days after the stem cell injection. This demonstrates that human neural stem cells that we grow and transplant into the brain parenchyma survive and stably integrate within the brain for at least 2 years and potentially for many years. In the right panel, another human neural stem cell line was genetically engineered to overexpress a marker protein, GFP, and injected into the spinal cord of nude rats. Nude rats are genetically immunodeficient, allowing for human cells to survive without graft rejection. These images were obtained 6 months after the stem cell injection. Each green dot you see here is a human cell expressing the transgene. This figure illustrates quantitative cell survival, stable and robust expression of the transgene, and widespread migration of the cells. Next, I would like to describe the key progress we have made to date. First, we have optimized the vector to maximize the level of enzyme expression inside the cells, secretion out of the cells, as well as the enzyme uptake by the neighboring cells. On the left panel, the enzyme activity in native human neural stem cells is rather low, and the secreted activity is quite low. To enhance the enzyme activity, we tested a lentiviral vector expressing the natural human SGSH coding sequence in a human neural stem cell line. This increased the overall expression level several-fold and increased the secreted enzyme amount. However, this level is still too low to be therapeutic. Next, we modified the human SGSH coding sequence to increase its expression level as well as the lentiviral vector. This resulted in several hundred-fold increase in the enzyme activity overall compared to the native neural stem cell line. About 50% of that enzyme is secreted out of the cells. About 50% of that can be taken up by neighboring cells. We believe that this level is potentially therapeutic. We next tested the enzyme's stability at various temperatures in culture. Once transplanted, the cells will continuously pump out the enzyme, which will mix into the brain's interstitial fluid and reach an equilibrium. This steady-state enzyme level will be available to be continuously taken up by the neighboring cells and digest away the accumulated heparan sulfate. At 37 degrees, the secreted enzyme maintained 95% of its activity for about 10 days. During this period, the enzyme will be continuously taken up by neighboring cells, and its activity will accumulate as illustrated by the graph. This level of enzyme secretion is sufficient to be potentially therapeutic. Infusing about 1.2 billion cells in 20 ml of volume will achieve about 25% of the normal enzyme activity level across the whole brain, which we believe will be therapeutic. So how do we distribute 1.2 billion cells inside the brain? We capitalize on the cell's property to travel fast on myelinated surface of white matter tracts. They are like the highways of the brain. Using these long cannula, schematically shown here, which are filled with the cells that traverse the brain. The cells are squirted out as the cannula is being retracted out of the brain slowly. The cannula trajectories are chosen so that the cells are placed near the major white matter tracts, which are illustrated here as vertical lines. We conceive about 8 cannula tracts, 4 on each hemisphere, to infuse the 1.2 billion cells in 20 ml total. The cannula trajectories are mapped preoperatively, as shown here, using standard neurosurgical procedures. These coordinates are then embedded into a custom stereotaxic platform, which is fabricated for each individual subject's skull and brain dimensions. This platform is used to hold the cannula steady and in precise trajectory. The platform is FDA approved. Using the clinically intended neurosurgical procedures and devices, we tested a range of infusion volumes in normal healthy dogs to find the maximum feasible volume. It was about 10% of the total brain volume. Thus, at about 1,500 ml for human adult brain volume, which is stable from age 5, the clinically planned 20 ml represents about 13% of the maximum feasible volume, with a safety margin of error of about 7.5 times. This is a good margin for first in-human trial. We also tested the cell dose range in normal healthy dogs, demonstrated that infusion of 1.2 billion cells, the clinically intended human dose, in dog brain, which is about 15 times smaller than human brain, was feasible and safe, which suggests a safety margin of error of 15 times, which is quite acceptable for first-in-human clinical trial. Shown here on the photo on the left is the actual cell infusion surgery on a dog subject. All components shown are clinically intended devices that are either already FDA approved or FDA exempt. The white sticks coming out of the head are the guides that hold each cannula and used to drive the cannula to the planned depth of the brain. These guides, in turn, are held on to the prefabricated stereotaxic platform that I described earlier. The cell suspension is loaded into regular disposable syringes, which are mounted into infusion pumps and connected to the infusion cannula by long nylon tubing. The cell suspension is pumped by the infusion pump. So there are 8 cannulas, 8 guides, and 8 infusion pumps. There are 2 neurosurgeons operating the cannulas and 1 assistant who is operating the infusion pumps. The total surgery time was about 4 hours. Now we put the dogs in 8 weeks of pharmacological immune suppression to allow the human cells to survive. The postmortem section here are stained with an antibody that recognizes only human cells. Each blue dot you see here is a human cell. Over the next 12 months, these cells will expand to about 6 times the original cell number and spread throughout the brain during this time. This is our current status. The green arrows indicate completed steps. White arrow indicate steps yet to be initiated. So using using research grade stem cells and research grade lentiviral vectors, we optimize the expression of the enzyme expression in the cells and out of the cells. We characterize them carefully. We tested the cells, the feasibility of the concept in mouse as well as dogs. We tested the clinical delivery device and procedures. We are now ready to make the actual GMP cell line for clinical trial. We have the recombinant enzyme in lentiviral vector in GMP grade ready. We are just starting to make the cell line of human neural stem cells. By early next year, I expect to have a GMP grade clinical lot of the final cell line overexpressing the enzyme. This clinical lot will then be tested in a series of IND-enabling non-clinical studies, which will, together with a clinical study protocol, will form the IND portfolio. I would like to acknowledge my collaborators thus far. The neurosurgery was performed by Dr. Nicholas Bullis and his team out of Emory University Department of Neurosurgery. Neurosurgical device was provided by FHC, who will also provide the devices for the clinical trials. The initial dog study was conducted at the University of Pennsylvania under Dr. Charles Beat. The funding was provided by Cure Sanfilippo Foundation. Thank you very much for your attention.
Okay, first of all, I would like to thank the Cure Sanfilippo Foundation, Cara, Glenn, all the patient families and the sponsor of this Congress, and also thanks for the opportunity to present our recent results, identified the CNS drug fluoxetine as a potential novel therapeutics for mucopolysaccharidosis type 3A. Next slide. So basically, the lack of SGSH promotes the primary storage of heparan sulfate of glucosam of GAGs that triggers secondary and tertiary storage, leading to a global lysosomal dysfunction. Global lysosomal dysfunction alters several secondary cascades like autophagy, and finally, these alterations are deleterious for the cells, in particular for neural cells, and promotes neurodegeneration. So based in this rationale, we thought that the potentiation of lysosomal activity may lead to an amelioration of the MPS 3A phenotype. So in order to do so, we develop an in vitro cell-based assays using high-content imaging approaches, where we can exogenously add a substrate that is specifically degraded within the lysosome and is fluorescent upon lysosomal mediated degradation, the DQBSA assay. So indeed, we found that in MPS 3A mouse embryonic fibroblasts, this assay show a reduction in lysosomal capacity to degrade these substrates, indicating that our hypothesis was correct, and the primary storage probably induced a global lysosomal dysfunction. Next slide. So by using these cell-based assays, we decide to survey a collection of FDA-approved drugs. In particular, we use the Presswick Chemical Library that contain 1,500 compounds that are clinically approved, and in order to identify compounds that are able to induce or to boost DQBSA degradation through the induction of lysosomal function. So among the different hits that we identified, we found the central nervous system agent fluoxetine, that is one of the most prescribed drugs to treat depression in adults and also in pediatric age. As we can see in the image on the right side, basically fluoxetine is able to promote or induce lysosomal activity in a dose response manner. Next slide. So interestingly, seeking for the mechanism of action of fluoxetine, we found that this compound was ineffective in cells depleted of the transcription factor TFEB that we already or previously showed is involved in the activation of global lysosomal functions, including autophagy and lysosomal exocytosis. So this data suggests us that at least in part, fluoxetine may mediate if induction of lysosomal function through the activation of the transcription factor E-beta TFEB. Interestingly, when we treat TFEB GFP cells with fluoxetine, we also observed that fluoxetine was able to induce the activation of TFEB through the induction of TFEB nuclear translocation. And in agreement with the activation of this transcription factor, we also identify an elevation of its transcriptional activity by looking at specific TFEB targets. In the right side, instead, we started to look for TFEB-mediated function that, as I say, are basically the induction of lysosomal biogenesis, lysosomal exocytosis, and autophagy. Looking at the autophagy pathway, we basically found that fluoxetine is able to induce autophagosome markers like LC3 and also reduce autophagic substrate like P62. Indeed, the action of fluoxetine is the promotion or the induction of autophagic flux, as we confirm by the increase in the fusion of autophagosome with lysosome using the LC3 tandem assay. Next slide. Then we look for the morphology of the lysosomal compartment, and we found that fluoxetine is able to relocalize the lysosomes in the close proximity of the plasma membrane, suggesting that fluoxetine is able to induce another TFEB-mediated pathway, which is lysosomal exocytosis. This was confirmed also by electron microscopy experiments where you can see that immunoglobuline staining lysosome are in the close proximity of the plasma membrane, suggesting that fluoxetine can induce the relocation and most likely the fusion of the lysosome with the plasma membrane to release their content. This was actually demonstrated by measuring lysosomal enzymes in the extracellular media of cells treated with the solvent or the carrier, the MSO, or with fluoxetine. So in these plots, you can see that fluoxetine is able to promote the secretion of cathepsin D, a lysosomal enzyme. However, there is no secretion of cytosolic enzymes like LDH that exclude the possibility that fluoxetine induce plasma membrane permeabilization. So these results indicates that fluoxetine may activate lysosomal exocytosis. In this regard, we tested whether by immunofluorescence whether the heparan sulfate that accumulates within the lysosome of MPS 3A-MEF could be induced by this mechanism, by lysosomal exocytosis, and secreted outside the cell. In these pictures, you can see that the treatment with fluoxetine relocalize heparan sulfate, lysosomal heparan sulfate, in the close proximity of the plasma membrane. indeed demonstrating that fluoxetine is able to induce lysosomal exocytosis. I have to say that several years ago, we demonstrate that the lysosomal exocytosis is a potential mechanism to promote clearance in several lysosomal storage disorders, including mucopolysaccharidosis and Batten disease. Next slide. So we decide to generate a proof of principle in vivo by using a spontaneous MPS IIIA mouse model. So we basically treat 6 months mutant mice with vehicle or 10 milligram kilo of fluoxetine, and we perform this treatment for 8 months. First of all, we collect mainly 2 tissues, the brain and the liver, in order to look for the effect of fluoxetine in the central nervous system, but also in peripheral organs. And we found that, first of all, fluoxetine was able to induce TFEP target genes in both tissues, in brain and liver, and also promotes autophagy, as we found in vitro experiments. Next slide. Interestingly, in brain and liver, the treatment with fluoxetine was able to reduce the vaporization that we measure by looking at the levels of the lysosomal membrane protein LAM1, and we do so by immunohistochemistry, immunofluorescence in these 2 organs, but also by electron microscopy. In the electron microscopy studies, we found that the size of the lysosome in mice treated with fluoxetine was significantly reduced, indicating that the fluoxetine-mediated clearance effect also promotes lysosomal biogenesis. Next slide. So, as I said before, our rationale was based in the evidence that primary storage induce the pathogenesis of MPS. So we test whether fluoxetine was able to reduce glycosaminoglycan content in mice treated with fluoxetine. And we found, by using Alcian blue staining in the left panel, and also the BlitzScan assay to quantify GAGs in the right panels, tissues from MPS IIIA mice treated with fluoxetine, and we found a significant reduction of heparan sulfate using both strategies. Indeed, we also identified by electron microscopy the reduction of the lysosomal compartment and the increase in the frequency of lysosome in the close proximity of the plasma membrane, indicating again that most likely fluoxetine can act through the activation of lysosomal exocytosis. Next slide. So finally, we also look for other markers or other pathological markers or hallmarks of MPS IIIA. So we look at the inflammation, status of liver in the left panel, and by looking at the inflammation marker CD68, we found a significant reduction of this marker in mice treated with fluoxetine, indicating a reduction of inflammation. In addition to that, we look by immunohistochemistry experiments the levels of the pathological marker alpha-synuclein in the cortex of untreated and treated mice, and we found a reduction in the phosphorylated form of alpha-synuclein after fluoxetine treatment. Similarly, we also observe a reduction of polyubiquitinated proteins, so proteins that aggregate during the neurodegeneration process, and we found a significant reduction of this polyubiquitinated protein in fluoxetine treated mice. So this data suggests that fluoxetine was able to reduce the neurodegeneration phenotype in these mice. Finally, we perform a functional assay by using the contextual conditional test, a behavioral test, that in general indicates that the MPS IIIA fail in the contextual fair conditioning, and the treatment with fluoxetine show a reduction or an amelioration of this test in mice treated with fluoxetine, suggesting that fluoxetine may improve the cognitive behavior of these mice. Next slide. So basically, by using a high-content imaging assay that was based in the measuring of the lysosomal degradative capacity of MPS IIIA cells, we identified the FDA-approved drugs fluoxetine and also the mechanism of action of this compound. So basically, we think that fluoxetine is able to induce clearance of glycosaminoglycans and ameliorates the phenotype in vitro and in vivo of MPS cells by promoting the translocation of the transcription factor A-beta that induce lysosomal biogenesis, autophagy, and lysosomal exocytosis. Next slide. So if you are more interested or interested in getting inside this study, this result was recently published in Molecular Therapy. Next slide. And now I will pass the word to Antonella Capuozzo, the first author in this paper, that we also make an update of our approach of drug combination in mucopolysaccharidosis type 3A and 3B. Please, Antonella.
Good morning. As Diego said, I will give just a brief update about the ongoing study. So next slide. By the primary screening by which we identify fluoxetine, we also found other hit compound belonging to several drug classes, among which we paid also attention on desloratadine, which is a sequel generation of antihistamine. And we decided to test together these two drugs in order to achieve a synergic effect. So meaning a high therapeutic approach with lower doses of the drugs and so less side effect. Moreover, these two drugs have also different tissue tropism, which can help to reach more impaired tissues. In particular, fluoxetine has as main target the brain, whereas desloratadine can reach more the peripheral tissues. Next slide. So we tested these two drugs in MPS IIIA mouse embryonic fibroblasts in our primary screening, the DCBSA assay, in a dose-response format. In particular, we treated the cells with a scalar dose of fluoxetine with or without a fixed concentration of desloratadine and vice versa. In both cases, we found a reduction of the EC50 value from 3 macromolar about from 1 macromolar without affecting the viability, suggesting that these two drugs can act together on the lysosomal phenotype, on improving the lysosomal phenotype. Next slide. We also move to MPS IIIB patients' fibroblasts, and we characterize 5 cell lines with different genetic background using some key experiment. In particular, by the DCBSA assay, we found that more or less all these cell lines have an impairment in the degradation of DCBSA, as well as there is an accumulation of the autophagy marker P62 evaluated by the immunoblot. Next slide. So we tested again this drug combination on this cell line and in dose response format, and we found that in all cases the combination of the two drugs are more effective than the drug alone. Next slide. So actually we are treating MPS III mice with a drug combination of fluoxetine and desloratadine and with the drugs alone. And at the end of July, we will collect the tissue, the brain and the peripheric tissue like the liver, in order to evaluate the efficacy of this strategy in vivo. And in particular, we will perform some immunohistochemical and biochemical approach to evaluate several pathological readouts like the accumulation of the primary storage of the glycosaminoglycans, the autophagy status, and the inflammation. So next slide. So we can conclude that the combination of fluoxetine and desloratadine produces synergic effect in MPS III and MPS IIIB cellular model, and now we are waiting for the in vivo results on MPS IIIA mice. Thank you, and now it's a pleasure for me to introduce Professor Giancarlo Parenti, which will be involved in the clinical study.
Yes, thank you. Can we move to the next slide, please? Yeah. So I will only say a few words about the potential of a clinical applications of fluoxetine. You have seen the results by Diego and Antonella. And of course, there is a potential for clinical translation. The advantage of fluoxetine is that fluoxetine is an approved drug. So you don't need very long preclinical studies to move into clinical evaluation. In principle, you can buy fluoxetine and start with this treatment. You only need a doctor that prescribes this drug. On the other hand, we don't want to treat patients with a drug if its effect is not proven, if it's a drug that is not useful or beneficial for patients. So the best option is to start a formal evaluation of such a drug, to design a clinical trial, to define what you want to see, what are the endpoints of this treatment, what you expect as a beneficial effect of this drug. Perhaps you have to identify several centers participating in this evaluation, this clinical evaluation, because this allows you to get more patients, to have more reliable results. And then we have to go through regulatory procedures that are very important to protect patients. Next slide, please. So the proposal is to use a standardized treatment protocol. You have to use in every patient the same dose, the same timing, the same treatment. You have to standardize also the evaluation protocols. You need the same tests that are done in the same way in all patients. Here you see there are some proposals like behavioral scales, biochemistry, GAG excretion in urines and other body fluids, and you have to monitor potential undesired events of this drug. And what is really important is to have a baseline. You cannot start treatment and see, say that there is some improvement. You have to compare the results of the same test before and after the treatment. Next, please. So everything has to be done according to good clinical practice. And good clinical practice means protecting patients, protecting from procedures that are too invasive, that are not necessary, and also means that you will end up with reliable results. You will know whether a certain drug is effective or not. Actually, this is something that we are currently doing with another drug. You will hear from Elvria DeLeonibus later in the session about another drug that has some potential for clinical translation. We are trying to start a clinical evaluation, a clinical trial with the help of the Italian Sanfilippo Association. And we have recruited more centers. We are trying to recruit patients. This is what we should do with any drug that we want to test. Perhaps a proposal, and I will stop with this proposal. No, not. We can go back to, yeah, thank you. I think we are running late with the slides, so I will stop here. Just proposing to put all our efforts together, different groups, perhaps we can identify centers throughout Europe or in other countries, and define all together a certain protocol, certain types of evaluations, certain endpoints, and then try to coordinate our efforts in order to have results that are reliable and are useful also for other patients, not only for those who are included in the trial. And I will stop here and thank also on behalf of the other speakers for your attention. Thank you.
Thank you. Thank you to everyone for those really wonderful talks. We have just a few minutes now for Q&A and have gotten so many wonderful questions. I will try to address as many as we can, but I know we won't get to them all. So we'll send them to you afterwards and to answer. We will start with Dr. Pavone. Could you comment on the effects of NK1 on lifespan and learning or memory in the mice, if that was explored?
Yeah, actually, this is the object of the project that Sanfilippo funded to us, and so the results, the study are ongoing. So Cara knows that we started already, but we do not have yet the results on the memory, learning, and the lifespan. So we are completing this during the project.
Thank you. And your intended route of human delivery, just kind of looking forward, what would you anticipate that delivery mechanism to be? Someone asked if that was the IV.
Yeah, intravenous in human.
Okay.
We hypothesize that intravenous would be the best for human. And then they asked me also about MPS 3C, maybe, yes, or 3A. So maybe it could be a good idea to try also on this. Mm-hmm. Okay. And that's it.
Thank you. Okay, Dr. Joe, we have a couple of questions for you. And the first one would be, how do you know how long the implanted cells survive, and do they differentiate into different cell types in the brain?
We know that from 2 clinical trials we conducted. One is in stroke. We've done about 30 patients in phase 1, phase 2. And nice thing about chronic stroke, as I illustrated in my slide, that we can see the infarct and we can see the graft survival. And we've done 24 months follow-up in all the subjects so far. So we know that that's at least how long the cells survive. In the second example, we also done clinical trials in ALS patients. And some of those ALS patients in our trial have died from the disease and had given us the opportunity to do the autopsy of their spinal cord in the transplant area. And we were able to identify the transplanted cells a couple of years after the transplantation. And the last question of differentiation, yes, these are tri-potential neural stem cells that differentiate into neurons, astrocytes, and oligodendrocytes.
Thank you, Dr. Joe. Dr. Medina and team, we have some questions about the potential side effects or concern of long-term or permanent TFEB activation in general and in non-target tissues.
Okay, so in principle, this is something that have to be clearly analyzed in both preclinical and potential safety studies in phase 1, phase 2 clinical trials. The nice thing is that fluoxetine could be also used in treatments that are not necessarily continuous. So you have to remember that you can stop the treatment with fluoxetine in principle if our preclinical studies indicates that is enough a boosting of TFEP activation to promote clearance and clean CNS and other peripheral tissues by lysosomal exocytosis, for instance. Okay, so this is something that we will study in preclinical models, but it could be also very interesting to have this data in phase 1, phase 2 safety clinical trials.
Okay, and you know, certainly there's a lot of human and even pediatric data on the use of fluoxetine, just general safety, not maybe particularly looking at this mechanism, but it has, you know, had with
this is a very important comment, because there are thousands of people that have been using fluoxetine. It could be very interesting also try to contact physicians in order to identify potential secondary effects that we could in some way link to the activation of lysosomal and autophagic pathways.
I think this links into the next question is, what would be the human equivalent dose of where you found efficacy in the mice? And, you know, is that a feasible human dose?
Yeah, in general, in humans, the dose is like 10 milligrams per day. Okay, so in mice we have to use a higher dose because the half-life of fluoxetine in mice is shorter than in humans. So this is also the reason why a phase 1, phase 2 safety studies could be also useful to make an escalation of the right fluoxetine dose in kids. You know?
Okay, wonderful. That's all the time we have for questions. We will share any additional questions that come in with you all afterwards. Thank you so much. This was really wonderful.
Thank you.
Thank you. Thank you, Cara. Bye-bye.
Bye. Thank you. Okay, we are going to go straight into our next session. And our next session is entitled Dopamine, Autism, and ABA Therapy in Sanfilippo. And we are thrilled to have with us today Dr. Elvria DeLeonibus, Group Leader of Neurogenetics at Telethon Institute of Genetics and Medicine in Naples, Italy, and followed by Suzanne Moore, Clinical Director at Bardin Behavioral Health in Columbia, South Carolina. Welcome to you both. And Dr. DeLeonibus, take it away.
Thank you, Cara. Thank you, and Glenn, for inviting me in this such an interesting meeting, and for all the effort to put it up. So, as Cara said, we are studying this. From 2007, this autistic behavior, which is the very early stage. Next slide, which is the very early stage, or the way Sanfilippo manifests. So I would just to acknowledge the role of Maria De Risi, who co-lead all the projects with me. So when I started to study these lysosomal storage disorders, in particular Sanfilippo, all the attention was on the last stage, when the neurodegeneration occurs and there is the lysosomal dysfunction, and you get that the children lose all their cognitive ability and motor ability they have acquired during their early life. But then studying the clinical course, we immediately realized that there was this really strange manifestation for a neurodegenerative disorder, which was basically a neurodevelopmental disorder. So all these autistic-like behaviors, like hyperactivity, which can be really invalidating, self-aggressive behavior, and other types of symptoms, which reminds more psychosis or autism in children. So we were triggered by which was the cause of this early occurring, next slide, early occurring manifestation. To do this, we had first, since we work in mice, to identify behavioral phenotypes which are similar or which can model the human pathology in mice. We focused on 2-month-old MPS III mice because we know from the studies and the collaboration together with Alessandro Fraldi that at this stage, these mice didn't have the lysosomal dysfunction. So this is a very early stage in terms of lysosomal dysfunction. And we found indeed that this early stage hyperactivity, this is an open field, and you put the mouse in this arena, they like the novelty, so they explore. But as you can see, the MPS III mice are really hyperactive. And then you can evaluate in mice stereotyped behavior. I'm not going into detail, but you have specific behaviors, and you see they are also increased at this stage. And then we also evaluated social interaction using these 2 tasks. Basically, in this task, in this where you see written the loser, you have 2 mice, a knockout and wild type mice, so an MPS IIIA. And as you can see, the MPS IIIA mice tend to retract when they confront with the wild type. So they are defined as loser, and this is considered as an impairment in the social interaction. And if you allow them to explore a T maze, and then they can choose between a conspecific, which is here, it's CD1, and a new object, usually wild type animals, so normal animals prefer the conspecific, while MPS IIIA mice tend to not show this preference. So we were, quite happy to have these endophenotypes at this stage, so these behavioral phenotypes, because they allow to look for the next slide, for which are the disease mechanisms underlying these autistic-like behaviors, but also to develop, which is also necessary to develop specific treatments for these autistic-like symptoms. So to study the neurobiological basis, next slide, we we focused our attention on the dopaminergic system. Why this? Because all the drugs that are used to treat these children, which are antipsychotics, like risperidone or haloperidol, are dopaminergic drugs. What is the dopaminergic system? The dopaminergic system is a neurotransmitter system, so a biochemical substance, which is produced in this area here. It's written like VTA and SNA on the right, where you see the brain of a mouse brain. And when it's produced, it's released in many brain regions, including the hippocampus, which regulates memory, prefrontal cortex, which regulates our executive function, and then the striatum here. In the striatum, it regulates hyperactivity, for instance, stereotype behavior, but also social interaction. Now, if you consider that in Parkinson's disease, which is an age-related neurodegenerative disorders, you have a degeneration of this system. You can see here the brain where the cells die, and these patients saw bradykinesia, so reduced movement, but also slowness in thinking and many other cognitive impairment. And the fact that in children with MPS IIIA, when you give risperidone, so drugs that act on this dopaminergic system, not only you don't get the beneficial effect, or at least they seem not so as much as effective as in iatrogenic autism, for instance, but they also, you get more extrapyramidal side effects. What are extrapyramidal side effects? I like catatonia or tremor. So behavioral symptoms that remind Parkinson's disease suggested us that this system might be altered in these children. Next slide. So we made a working hypothesis suggesting that this initial hyperactivity, stereotype behavior, and social impairment or autistic-like symptoms might be due to an hyperdopaminergia in the brain of Sanfilippo. Why? If you take, for instance, amphetamine that increase the amount of dopamine in the brain, or you take cocaine, you become hyperactive, you show stereotypes, and also you show social interaction impairment. When this amount of dopamine is reduced, like I showed you in Parkinson's disease, then like due to the neuronal loss, you get hypoactivity, neuromuscular deficit, and cognitive impairment. So basically, a change which in upregulation or downregulation in this system might support some of the symptoms that you see in the children in the early stage and in the late stage. So next slide. So together with Maria, we looked in the brain of MPS III mice at a very early stage. Here is during the embryonic life when the dopaminergic system starts to originate. And as you can see on the right, you see that the number of TH is the enzyme that produces dopamine cell, are increased. And also the proliferation of this cell is increased. And so this was, quite surprising because in a neurodegenerative disorder, you have an increase in the number of cells. And we confirmed this also in 2-month-old mice. So you have this increase in the number of cells during the embryonic life, which is maintained also during adulthood. Now, next slide. We wanted to know why this you have this number of increased cells. At this early stage, this is an electron microscopy analysis of the cortex of MPS III mice produced in the lab of Alessandro Fraldi. And as you can see, at 3 months, there is no lysosomal dysfunction. So at this very early stage, the lysosomal dysfunction is still not, the lysosome can still compensate for the storage of heparan sulfate. So we focused our attention on the possible alteration of the function of the heparan sulfate. You know almost everything now on the physiological function of the heparan sulfate because Professor Pavone already introduced this. So we tested, next slide. So we wanted to confirm in our mice, so in MPS III mice, we wanted to confirm that the function of the heparan sulfate was altered using this assay. There were already previous evidence showing that you not only have more heparan sulfate in the brain of Sanfilippo or lysosomal storage disorder, not in particular on MPS IIIA, but the heparan sulfate that is accumulating also lose its function as co-receptor, for instance, of FGF2. So through this assay, we showed that the heparan sulfate extracted from the brain of MPS III mice was not functional, so was not properly make the FGF2 function. Because, next slide, as Professor Pavone said, the functional heparan sulfate is necessary to bind, to allow FGF2, which is a fibroblast growth factor, to bind its receptor, which is FGFR1. So, the FGF2 is crucial for the development of the dopaminergic system. Next slide. In this, for instance, in this paper, they showed that if you have a knockout a model for FGF2, you have mice that recapitulates the brain or the dopaminergic system that we see in our MPS III mice. They have more dopaminergic cells. So this is just an indirect indication that when this signaling between heparan sulfate maybe and FGF2 is not working, you may have hyperdopaminergia and an increase in the expansion domain of this system, which is the dopaminergic system. Now, we wanted to prove this in vitro. Next slide. So we used a cellular model generated by Ilenia Monfragola in the lab of Andrea Ballabio, made with CRISPR-Cas9. And as you can see here, you have wild-type cell and MPS IIIA cell. This is a neuroblastoma cell line. And when you remove the, you have the MPS IIIA model, you see that the amount of cell, even if when you put them in the dish, you put the same number, after 4 days, you get a huge amount of cells, so a huge proliferation of dopaminergic cell. to test whether this proliferation was dependent on the amount of heparan sulfate, dysfunctional heparan sulfate accumulating in this model, we added to the medium functional heparan sulfate. And when you add functional heparan sulfate, here written like HS 25 microgram or 40 milligram, you see that the number of cells in the MPS IIIA dish is reduced, meaning that this one might be one of the mechanisms contributing to this hyperproliferation, so to this hyperdopaminergia. Next slide. So, I mean, the set of data we have collected are more or less in line with the view that was taken also by Professor Pavone, but also by other scientists in the field like Professor Esco, suggesting that we have not only a lysosomal dysfunction in this pathology, and this is not the only cause of all the behavioral phenotype that we see, but we have a loss of function or an altered function of physiological function of heparan sulfate that might be responsible for the neurodevelopmental disorder that we see in these children. And when we design therapies, we have to take into account both disease pathways. Next slide. This is, I mean, important. This finding is also important because in iatrogenic autism, so in form of autism which are not associated to Sanfilippo or altered gene mutation in heparan sulfate, it has been reported. This is a mouse model, but also in humans, it has been reported a loss of heparan sulfate or alteration in heparan sulfate signaling. So this finding might have also a more general impact for understanding the mechanism leading to some of the behavioral manifestation associated to autism. Next slide. But then we wanted to know which is the neurochemical imbalance in this dopaminergic system, because if you want to help and to improve the quality of the life of the patients, you have to act at the level of the synapse. So you have to act with drugs on the dopaminergic system. Next slide. So we evaluated together with Maria. This is a dopaminergic synapse. This is a little bit complicated for people who are not in the field, but this is a normal synapse, and the dopamine is packaged in these blue dots. And when it's synthesized in the synapse, this dopamine is released in the synaptic space. Now, what determines the amount of dopamine that you have in the synaptic space is not only how much cell do you have, and we have seen that in MPS IIIA mice, we see more cells or more dopamine, but it's also dependent on these D2 receptors here in red in the presynaptic space, because these D2 receptors have an inhibitory action on the release of dopamine. So they can control, it's like a DIM, they can control the amount of dopamine that is released. And then the net effect that you have on behavior is dependent on the action of this dopamine on two receptor classes, which have opposite effect on an intracellular pathway and acting on D1 receptor and D2 receptor. Now, what we have found in MPS IIIA synapse in this striatum, we found that they have more dopamine, but they have less inhibitory control in the release of dopamine. So they synthesize more and they are less able to inhibit the release of this dopamine. So it's like an outflow, a huge outflow of the amount of dopamine. And then as a net effect at the postsynaptic level, you have an increase of this D1 pathway to the detriment of this D2 pathway. So there is an imbalance. So our goal at this point was either to reduce the amount of dopamine that is released to manage the symptoms, or to really reestablish the balance between this D1 and D2 pathway. Next slide. Now, we have started from was the clinical evidence that risperidone, which blocks the D2 pathway, is not working and gives more than expected side effect. Next slide. So according to our data, why risperidone should do this? Because if this D2 pathway is already underfunctional in MPS IIIA, risperidone is going to block this both at the postsynaptic side and at the presynaptic side. So it worsens the situation because it increases even more the amount of dopamine by further blocking the inhibition of the release, but also is further deregulating this signaling. And indeed, when we give give haloperidol, which is an antipsychotic similar to risperidone as a mechanism of action, you see that you reduce only hyperactivity, here the figure B. But all the other behaviors are not rescued by this drug. But this reduction in hyperactivity is really functional. We are recovering something, or we are producing side effect. Next slide. Because when you increase the dose of haloperidol, what you see, what I told you, is a cataleptic effect or extrapyramidal symptoms. So meaning the mice becomes immobilized and the children become a little bit catatonic, not responsive. And this is something that parents do not want, but also clinicians do not want. So when you look at the MPS IIIA mice, you see that the cataleptic effect of haloperidol, as for risperidone in children, are much more evident as compared to the wild-type animals. So this is not the good treatment. So we tried another approach, next slide, which was based on reducing this hyperactivation of the D1 pathway with an antagonist that specifically act on this D1, so rebalance between D1 and D2 signaling. And this antagonist works first because it rescues all the behavioral manifestation, but also because, it's effective in rebalancing also the molecular mechanism that I'm not going to show you here, but it's published in the paper. But this drug is not in clinic and has an half-life which is very short. So it makes it a little bit difficult to use it for regulating the clinical manifestation all along the day. So we looked for another drug, which in our idea, next slide, could be safer and could be also important for continue the treatment also during, according when the lysosomal dysfunction could start. Because you need a drug that can adapt to the level of dopamine that is changing in a predictable way during the development progression. Next slide. So I want to thank you for your attention. My group in Naples, we are also sparse in Italy, which is the group in Naples is focusing on this project and also other projects, also all related to Sanfilippo. A huge thank to our collaborators, but a special thank to all the families and the association of families who are supporting our research and permitted all this. Thank you.
Hello, everybody. I wanted to say thank you so much for this opportunity to the Cure Sanfilippo Foundation. My name is Suzanne Moore. I'm a board-certified behavior analyst, and I am the current clinical director for Bardin Behavioral Health, and we provide ABA services in Columbia, South Carolina. You can go to the next slide. Thank you. And so just to touch on ABA a little bit, applied behavior analysis is the use of evidence-based practices and procedures to make significant changes to behaviors. Essentially, we are shaping behaviors, and it's really important to understand that we are trying to shape socially significant behaviors so that they mean something to our clients. We want to make sure that the things that we are shaping help to increase their quality of life and their communication skills. Next slide. Okay, so where do we start when we start to provide services in ABA? We start with an assessment, and that includes things like communication, daily living skills, physical, gross, and fine motor skills, problem behavior, and medical issues that might come up that would lead to problem behaviors, stakeholders, and collaboration and preferences. And I want to go over what each one of those things are. So let's talk about the assessments first. Next slide. So these are some of the assessments that I prefer to use, and there are tons of assessments out there that are validated that you can use, but specifically with individuals that may have more skill deficits or may have regression in skill deficits, I find these to be useful. And so there is the Essentials for Living, and the PEAK assessment, the Vineland-3, and then we have the VB MAP, which is the Verbal Behavior Milestones Assessment and Placement Program. And then we have the ISCA, which is the Interview Informed Synthesized Contingency Analysis. And I could go on for a very long time about each one of these assessments. And so I'm happy to answer questions on the side about these assessments if you have questions about how they could apply to an individual with Sanfilippo syndrome. I'm happy to address those. But for just case study reference for an individual that was 12 years old, now I use the Essentials for Living. I use the Vineland-3 and the VB map, and I've also used components of the ISCA and the practical functional assessment, which goes in line with the ISCA. Next slide. And so this is a graph that Cara was kind enough to give me to show a regression. This is a retrospective analysis by Berman et al. Of a cohort of 21 boys and 25 girls with MPS IIIA identified early and significant language developmental delay and hearing loss, followed by a rapid decline in cognitive skills and adaptive behavior after 3 years of age. The parents reported that the children had multiple behavioral and sleep difficulties during the period of cognitive decline. 2 prospective natural history studies confirmed these findings and explored the outcome measures most suitable for assessing changes in cognition in MPS IIIA patients. And so you can see that there's a very quick decline there. And I think that it's really important to see something like this as a provider, because you are going to be looking at a quick decline in communication and skills that are necessary for daily living, which means you may have a quick increase in problem behaviors as a result of those skill deficits. And I think that as we move in and provide assessment to individuals with these type of deficits, it's important to understand the level of frustration and the level of difficulty and challenges that we might face in helping an individual where they are at currently. It may be more difficult to assess those things. Next slide. Okay, so let's talk about communication. And I think when we're talking about communication, we need to be careful not just to think about vocal communication, although that is a main form of communication for many individuals. It is more difficult with individuals who have dementia, individuals who are autistic or have autism. And so we really want to look at all the different types of communications that we could have in this situation. So we could have eye gazes that we're looking at, communication devices, gestures, picture exchange communication systems, problem behavior, leading another person who knows the individual well. We want to assess what type of communication the individual is currently using and what will be in their best interest to use moving forward. And I think it's always important when you have somebody with a very low communication skills to have another person who is very familiar with them, at least in the beginning of stages of that treatment until the provider knows the individual well, and other people are available to provide that assistance. Next slide. And so daily living skills, and of course, this is just some of the examples on this chart here that I put, but there are many that you could look at. The part was a little more difficult to assess with an individual who is having more physical difficulty with some of these daily living skills. It is important to think about what stress we are putting on the client, and some of the behaviors may be more challenging due to medical problems such as hypotonia, muscle tone, and motor planning difficulties. I point out this because some kids have increased tone rather than low tone, so it may be something to think about as you're moving forward with the assessment in making sure that we're not pushing them to do something that they're not physically capable to do, and meeting them where they're at when we write those goals. So we want to look at increasing independence through approximations, and approximations are smaller steps that we take and reinforce to get to a terminal outcome. If the client is reaching for a cup, we reinforce that approximation and aim to increase the next level of independence. So if they're reaching for the cup, I love how you're reaching for that cup, and then perhaps I'll provide assistance at the elbow. So we're going to work to try to get them to be more independent, but understanding that might not be physically capable for that individual. And so making sure we write those goals to reflect that. Ultimately, we are trying to maintain skills, but if we are able to build skills, we want to make sure we're doing that and looking at things as they are possible and not discounting them because there is a physical limitation. Next slide. And so as we're looking to assess, we again want to look at the gross and fine motor, and we want to look at things such as walking, sitting, climbing stairs, and for fine motor, grabbing, holding objects, or pointing. But these skills are going to be increasingly important as a client gets older and the disease progresses, so that we can help them maintain that muscle tone and help them maintain the physical ability to do these as best they can. And things like pointing, can I isolate my index finger to point and touch a communication device? Can I push a button if I need to? Can I mash a Big Mac if I need to? Those are all really important skills to be able to communicate wants and needs, or even pain or sickness. Next slide. And so we also are going to assess problem behavior, and we are going to look at things such as flopping, tantrums, elopement, aggression, self-injury, and of course this list could be longer. These are just some examples of things that I see. Flopping is really important because it is a type of possible escape behavior, and it's really hard to tell when somebody can't communicate why they're escaping. So we want to make sure that we're always looking at that when we're assessing, especially we're assessing a new client. Why are they flopping? When are they flopping? What are they flopping right afterwards? And the same is true for tantrums, elopement, aggression, and self-injury. And of course, if there are more extreme behaviors, we are going to want to address those first, which means we're going to have to implement other assessments such as a functional analysis to determine the function of those behaviors. And that includes determining whether it is something that is being provoked by medical issues. Next slide. Okay, so let's talk about medical. And I think that the medical is really important, especially when we have clients who have Sanfilippo syndrome, or we have clients who have other disabilities that may have more medical issues. We want to look at their medication. You know, if they are taking something like haloperidol, it's important that we know that. And the reasoning for that is because it does cause behavioral manifestations such as the catatonic states, lower rates of communication. We want to make sure that if they miss a medication, that we know that, because that could lead to other behaviors that are not something that we're working to shape, but that we do have to work around when we're working on behavioral skills. Your physical disabilities, we really want to look at what physical disabilities are there, but how can we meet that individual and bring them to a level where they can have quality of life and access their daily living skills with as much independence as possible without it hurting them, without it stressing them. Illness, it really is important to look at the illness. If somebody has more issues with constipation, they may be less likely to interact during therapy. They're less likely to eat, which means that if food is something that is reinforcing to that individual, it's something that they might not be willing to work for. Feeding issues, if the individual does have feeding issues, you want to make sure that you know what those are and that you're working with the speech pathologist to understand the behaviors and whether it is something you can or cannot work on. And then sleep habits. Obviously, if we're not sleeping, we're going to have an increase in problem behavior that next day. And that could mean something as simple as not responding to somebody who's trying to work with you. That could be seen as a problem behavior, especially if we're working on those really important daily living skills. Next slide. And I think this is a really important piece in any case, but I also think it's really important in a situation where we're working with somebody who has medical needs, and we want to make sure that we are working with this as it is an ongoing process, because things we may try might not work. And I think as a provider or as a family, a teacher, OT, PT, physician's speech, it's really important to consider that with any case, but especially with a case where we have somebody with a degenerative disability. We want to make sure that we are collaborating with the stakeholders and making sure that we're talking to each other about what is or isn't working, and then changing those things when they don't work. And families are really good to talk to you about things that are not working at home, things that are not feasible, and sometimes you have to get a little creative. And so collaboration will help you through that process, especially when you're working with feeding, disorders, where we're working with stressful situations. You want to be working with the speech and counselor to make sure that your treatment is going along the same lines with the other stakeholders. You want to make sure that what you're doing in the home or at the clinic is not causing more stress to the individual or their family. Let's look at the next slide. Okay, so preferences. I think this sometimes is one of my favorite things to work on with clients, but you want to make sure that you're assessing preferences, looking at their ability to express preference, to express joy and non-preference. That's really important. You may have an individual who, if you present something to them, they might not respond, but that doesn't mean that they're not responding inside. So we want to make sure that we understand how the individual expresses preference so that we can determine what they do and don't like the most. And this may be a longer process. And so when you're doing that initial assessment, you do want to make sure you're collecting baseline on whether the individual is able to express those likes or dislikes. Does the client attend to things they once liked? So if the TV pops on, do they look up in the direction of the TV? Do they attend to the people around them? So if somebody walks in the room, do they look at that individual? You want to make sure you're really attending to those things during the assessment so that you can help shape that behavior. Those types of behaviors as an individual gets older become more important as they're in residential settings or having other providers push in that don't know them. You want to make sure that you understand what those preferences are so you can share it with other people. Another thing to think about in this type of assessment with the preferences is, do they turn their head? Do they flop? Do they throw toys on the ground, push things away? Are toys aversive to them? Is it not a preference for them? Perhaps preference for them is escape from interactions or escape from those toys. And, you know, keeping in mind that they're going through a lot of therapies. And so when those toys are being presented, perhaps they're having trials run on the toys and they're no longer toys to them, they're work. So you really want to make sure that you understand what those preferences are, what activities, people, things, smells, or places that individual likes or doesn't like. And if it is not clear what those things are presently, that is where you're having interviews with the parents and making sure that they're giving you that information about what an individual once liked and trying those things one thing at a time to see if they still prefer those items. Next slide. And so I wanted to give an example. This is a cumulative graph formatting for an individual who is 12 years old and is diagnosed with autism and Sanfilippo syndrome. And this is a really important graph to look at because this was something that I had to change the way I was graphing things. I do still graph using frequency graphs, but it is easier to see progress with cumulative graphs. So one of the considerations I've learned to account for through my experience and through training from experts in the field of ABA is that we must put aside our expectations and biases. We must consider that there is a presumed trauma, presumed behaviors that have been put on extinction or forgotten. An example is when my client sees their favorite show and has a brief moment where they smile or jump, I smile and jump with them. When the client looks at me, I smile to reinforce that I see them looking at me and I am here for them. Acknowledging eye gazes, acknowledging small indicators of interactions, and I continue to have small conversations and talk about what is happening in the shows, despite a lack of reciprocity in the conversation. These are pieces of treatment that are incredibly necessary when shaping behavior and using synthesized contingencies to ensure the client is happy, relaxed, and engaged, and ready for learning and retaining skills. And what I hope that you take from my presentation today is that children with Sanfilippo can make progress. The progress may not be what we expect, but it is necessary to consider that what we expect is based on what we know about typical development. Because we are not working with a child who is typically developing, we must meet them where they are at, determine barriers to quality of life and independence, and provide support to remove the barriers, enable the client's ability to pursue happiness and independence as much as possible. And to remember to be flexible in treatment and to remember to reinforce the small things and document them. And the small things matter and they add up to larger things and increase the likelihood of success. And I think that cumulative graphs can show that. And I really wanted you to see where we started, which was very low mandating with AAC device is at 4, and now we're up to 24. And this does vary from day to day, and I think that's really important to consider. is that when you have an individual with medical needs, you have an individual who may be losing sleep or maybe on different types of medications to address hyperactivity or some of the symptoms associated with autism and Sanfilippo syndrome, that you might have variability in manding. If an individual is not hungry, then there is no reason to mand for food because I'm not hungry. So you might have a lower count that day, or perhaps that individual is tired that day. Maybe they had several doctor's appointments and now they have this ABA appointment, and you might see a variability in the graph that looks like this. But you want to look at that slow ascending trend line. That is where we're aiming. Can we increase this across time? And shaping behavior can take a long time. And so that's important to consider. And as you are assessing your client, I highly recommend taking a baseline of behaviors that are indicators of happiness so that you can track that as well and ensure that your treatment is effective with the client and increasing their quality of life. And that, I think, is a really important piece to consider. Next slide. And so here are some resources that I wanted you all to have. And of course, these are some of the experts in the field of ABA that I have let guide my practice and the way that I interact with my clients. Essentials for Living is by Dr. McGreevy. And I highly recommend going to his site and checking out their assessment. It is targeted at all the different levels of ability and looking at where your client is right now and helping you to be able to shape their behaviors and finding a communication that is most appropriate for your client. And that is where I pulled the global communication system, thank you, that is was appropriate for my client. And the ISCA, which is the interview-informed synthesized contingency assessment, I highly recommend looking at the research behind that. Your client might not have intense problem behaviors, but the philosophy behind that assessment is spot on. I highly recommend checking that out and checking out Dr. Hanley's courses. And if you have feeding disorders and you're a behavior analyst, you may want to check out the feeding disorders information consultations by Dr. Piazza. And then, of course, a wealth of information on Sanfilippo syndrome is the Cure Sanfilippo Foundation website. And next slide is my contact information. Please feel free to reach out to me. I may take 24 to 48 hours to respond, but I look forward to questions. And thank you all so much for coming today and listening to me. And I wish you all a happy summer. Thank you so much.
Thank you, Ms. Moore and Dr. DeLeonibus for those really wonderful talks. We have a few questions and a few minutes to answer them. Okay, let's start with Dr. DeLeonibus. Do you think that a blood-brain barrier penetrating enzyme replacement therapy can degrade central nervous system heparan sulfate enough or such that it can improve the dopaminergic neurons and leading to improvement of autism? Like, would that be an expectation of, enzyme restorative approaches as well?
You mean postnatal?
Postnatally, yes. Can you restore the dopamine neurons postnatally?
Yeah, for sure. I mean, I think this finding of alteration already occurring during the embryonic life, if they are confirmed in the children, it means that any postnatal treatment might leave something that is not treated. So this can be enzyme replacement therapy or can be gene therapy. It's really depending on, because there is still a window to modify the development of the brain postnatally, because it's not complete after birth, immediately after birth. So now what we want to test, and we want to see whether we can really, acting in the early stage, we can modify these, let's say, development or enlargement of the dopaminergic system. So if enzyme replacement therapy would work or could cross the brain, or gene therapy, if it's done really immediately after life, we might have some window to prevent or at least to have less severe autistic-like maybe phenotypes. But if there is this prenatal component, maybe something would be left.
Could you comment on, the proposed perhaps future trial with aripiprazole, potential side effects and how they might be different than risperidone?
The question is for Suzanne?
The question is for you. I'm sorry, Dr. Dewey.
Okay. So the point is that aripiprazole has been, even in non-MPS3A, so in iatrogenic autism and so on, has been shown to have less extrapyramidal side effects as compared to risperidone. The important thing that we are seeing is that the dose that you use is extremely relevant. The dose used in clinic, the doses of these drugs used in clinic are usually very high, even in our models. And with any type of dopaminergic drug, you can have that, for instance, a low dose has pro-cognitive effect. When you increase the dose, you get side cognitive effect, so impairing cognitive effect. You think to amphetamine, you take amphetamine, you take a small dose, you are more attentive. You increase the dose, you become completely unattentive. So all the drugs that act on the dopaminergic system are really extremely sensitive to the dose used. In children that have a neurodegenerative disorder going on, you give risperidone, but think, you would ever give risperidone to a Parkinsonian patient or to an Alzheimer patient? Risperidone is not given. So when we give these drugs to children that have a neurodegenerative disorder, we have to think whether we are also acting on this other pathway and exaggerate what is already going on, which are extrapyramidal symptoms. They develop spontaneously. So I think that, aripiprazole at low dose, this is what we are testing at very low doses, might have more beneficial and less side effect as compared to risperidone. But this is something it's just based on our preclinical data. So we will see.
Okay, thank you. Thank you. The next question is for Ms. Moore. Knowing that families are regularly faced with educating their own providers about their child's rare condition, what are the key things that you recommend that families communicate specifically to an ABA therapist to help them optimize their approach to treating a child with Sanfilippo?
That's a really good question. And I think that communication is key. And I think that your understanding as a parent, as a family, of your child's communication is going to be really beneficial to share. So sharing things like, what do they gaze at? What do they look in the direction of? What causes them to smile? So perhaps there's a song that they loved many moons ago, and when it comes on, they smile. That's important. If food is reinforcing for them and it's not dangerous for them, like they're not shoveling and putting it in their mouth, those things are very important. And helping them to know when your child doesn't want something. So do they turn their head? When they don't want something, do they flop to the floor? Do they put their hand in their mouth? What are the indicators showing what they do and don't like? If we know what those things are, then we can help shape the behaviors using those reinforcements. If they don't like to be touched, that's really important. And also, what is their history with therapy? I think that was a really key benefit to me to know, what has happened in the past. Have they had, forced compliance? What have they been through? Have they had great therapy? Do they have great therapy at school? What does it look like? So that we're not competing with that, one, and two, so that we can know that, hey, this individual has a hard time with being touched, and we might want to not do that right away.
Thank you. Sure. Okay, thank you both. I know this has stimulated a lot of thought from everybody, and you're probably going to be getting lots of questions afterwards. Ms. Moore, I know the resources you provided are of great interest to the group, so we'll find a way to share those in the platform as well.
Sounds
Thank you
great.
both.
Thank you.
Have a great
Bye.
day, and we will move on to our next session. We are super excited to host some very special people in this next session, entitled Connecting Life and Science. And this is the first of 3 such sessions, one for subtypes A, B, and C, where we bring together 3 parents, a scientist, and a clinical doctor to share experiences and have open discussion. Our intention here is to offer participants and the audience a glimpse into each other's worlds, and that the interactions will bring new insights. In this session, we will focus on Sanfilippo type C. And joining us today are 3 amazing mothers who all have children with Sanfilippo type C. And I'll hand it over to you guys to introduce a little bit about yourselves, your children. Sybil, would you like to start?
Sure. Good morning. My name is Sybil Aros-Pearson. I'm the mother of Solomon Pearson. He's 13. He'll be 14 next month. And 2 girls, Simone, who is just turned 16, and Joanna, who's 26. Thanks for having me.
Thank you. Christi?
Hi, everybody. I am Christi, and my son is Levi. He'll be 24 this coming Monday. We are so excited about that. I live in Wisconsin, and I have another daughter who is she's superior. She's 27.
Great. Marissa?
Hi, guys. My name is Marissa DiCiaccio. My son is Connor Dobbin. He just turned 14. 2 weeks ago. We live in Pennsylvania, and he's got a typically developing brother named Keenan, who just turned 12 last month.
Fantastic. We're also fortunate to have with us Dr. Dingle and Dr. Badell-Graum. If you'll each just introduce yourselves briefly and tell us how you became connected with Sanfilippo syndrome.
Sure, I can go first. So I'm Dr. Dingle Sanchez. I'm at UT Southwestern. And I actually split my time between neurology and oncology over here. My main area of interest is actually, my main focus has been neurofibromatosis. But on the neurology side, I see a lot of rare disorders and have been involved in some research in rare disorders. We actually at UT Southwestern don't have any patients with type C, but we have a very robust gene therapy program, which is how we became involved with type C.
Thank you.
Hi, I'm Dr. Badell-Graum. So I'm currently a postdoctoral fellow at UC San Diego. I actually have done all my studies and all my work, master's and PhD and undergraduate studies in the UK, where I worked on lysosomal storage diseases. I mainly focused on drug development and drug screening for lysosomal storage disease. I worked on mainly Batten disease and Niemann-Pick. And then Dr. Stephanie Cherky here at UCSD basically brought me over to work with her on MPS IIIC, on gene therapy, hematopoietic stem cell transplant gene therapy approach. That's a brief background on me.
Thank you. So I'll help facilitate the discussion in the session, but I would encourage any of our panelists to jump in if a discussion point comes to mind or you have questions for each other. This is your session, so feel free. To just kind of kick off the discussion, I thought it would be nice to start with our mothers and ask them, when did you first start to have an inkling that something just wasn't right or going typically with your child? And when did that happen?
I can start off. I would say I honestly felt it kind of early on with Levi. My daughter, she was advancing very fast with a lot of things. I was a stay-at-home mom. I ran a home daycare, so I was doing a lot of educational things with the kids, teaching everything. And Levi was right there with everybody, playing and doing everything, but he just seemed to be not catching on with all the other children. I did compare him to my daughter and other kids at that time. He was maybe 2.5 3. So I went to the doctors and stuff like that, and everybody said no comparing. You know, he is a boy. Your daughter, you know, she's a girl, so of course they do things differently and learn things faster than boys. So I was kind of put on the back iron for that. So yeah, he was probably about, I think about 2.5, 3 years old when we first finally when I really saw something. And when everybody kept telling me, no, he's, you know, he'll catch up. But I knew deep down in as a mother that there was something more, not Sanfilippo, but I knew there was something more than just a delay.
Yeah, the gut feeling kind of.
Right.
Marissa, how about you?
Yeah, totally similar. I actually noticed right around a year, his very first, year pediatrician appointment, I brought it up in terms of his communication. Same exact thing. Oh, he's a boy, he's a summer birthday. They tend to be a little delayed. Then I kind of kept pushing, and we actually got accepted in an early intervention at 18 months. So I kept pushing, and I knew around 3.5, we went to genetics. Actually, nothing popped up because it was probably your basic screen. So I thought we were on the journey for autism. That's obviously what I thought we were working with. And then we finally got the autism diagnosis at 5.5. So I kind of knew from very early on that something was going on. I just, like Christi said, did not think it was Sanfilippo. I thought autism was our journey.
Yeah. Sybil, it's kind of a similar scenario?
Early on, he has a sister who's 2 years older, so I did the same comparison. But initially, because he could read at 3 and do basic math, we thought, oh, he's just bored. He's brilliant, right? We thought, so all those other diagnoses coupled with his pediatrician he was very old school and didn't believe in ADHD and autism. It was too early to tell. So in preschool, the teachers kept saying, you have to get him tested, but nobody, of course, wanted to say autism or ADHD. So then he went from Montessori, which you can individually do tasks, and he did those on his own. He just never wanted to do group play. He never wanted to do structure. So when he went to kindergarten, it was problematic because he went to public school and, it's a box. All the kids do this at this time. So he created these like exorbitant escape mechanisms and, still we got auditory processing disorder diagnosis initially, then we got ADHD. And then finally in first grade, he was put in the gifted class, and the time away to focus with the resource teacher said, take him to the resource center. We have these resource centers that are free resources to diagnose. And they gave us the autism diagnosis, and we were devastated, of course. But we thought, well, people with autism go on to be engineers and all these different things. And so they changed him to a school that had smaller classroom sizes. And there was just a sharp, immediate decline in his abilities. So we thought, that's when I really was like, okay, something more is going on, because with autism, you don't lose the skills per se. And all the research I did, nothing pointed to anything in regression outside of 2 years old, 18 months. So I could just never pinpoint it. So I changed pediatricians when he went to middle school, and he lost his toilet training abilities and just clearly couldn't read anymore. And he actually, before middle school, went back to his old school and ended up in a special ed class that was not part of the core curriculum. And the teachers that had assessed his IEP initially said, what did they do to him at the other school? I'm going to bring him up to par because he could read, what were they doing over there? And we were like, yeah, it's the teacher. So we always had an excuse for all these different things. They weren't challenging him, and he lost all these things. And then it just became evident when she was like, I'm trying, but, I don't know what's going on. And so I started asking, ABA therapy, what have you seen this before? And everybody was like, no. And so you can markedly see just the difference in the years. So eventually, when I changed pediatricians and I read an article about some markers in autism being genetic, and I said, can I go to genetics? And finally she said, okay, she wrote me the referral. I waited 6 months, and then we got the Sanfilippo diagnosis, and we were like, I just wish it was autism, So that's kind of our long journey. It was 5 or 6 years in the making to get there.
Yeah, pretty remarkable given that each of you, had that red flag that went up very, very early on, but, what's the message there to our providers?
Yeah,
Dr. Sanchez, how do you reflect on, hearing that?
Yeah, well, my heart goes out to you, of course. I think we very often, we hear that with other disorders very often. I just knew, the moms or the families say, I just knew something was off, something was different. I mean, in our defense, it's hard on our side because for every family that knows, there's 10 families that know and it doesn't turn into anything. So it's a little bit hard to know which kids to go full force sedating and doing invasive testing and so forth to look for something, versus just more watchful waiting. It's also a little difficult because in many cases, I mean, I think that even if y'all had been heard appropriately, I don't know that the clinician would have known the right test to find that diagnosis early on. And a little bit of it is waiting until a little bit later until other symptoms show up that tell you what tests to do. I think I'd be interested in hearing for each of you how the diagnosis was made, because usually I don't think it would be from the first test that gets sent. So was there something else that then popped up that told somebody what specific testing to do, like the urine testing, or was it a more broad genetic approach, or how did y'all finally get your diagnosis?
I'll go first. It was interesting. We took Connor while we were trying to get the autism diagnosis. We took him to a developmental pediatrician who had recommended genetics. So we did do genetics at 3.5 at CHOP, Children's Hospital of Philadelphia. But I think they did like just your standard, screening, so nothing popped up. So it took a while for us, and it wasn't until 5.5 when he went to kindergarten where we partnered with this kindergarten teacher, and she finally was seeing him with other children and communication, social skills, the whole 9 yards. We went back, we ended up getting an autism diagnosis. What was interesting is at in third grade, because we repeated him in kindergarten, so every 4 years our school district legally has to do a new psychological evaluation on him. So in third grade, they did it, and they called, I'm divorced, so Connor's dad, his name is Mike. So Mike and I went in just kind of thinking it was going to be a same old, like this is what we're seeing. And they said to us, when we tested him in kindergarten the first year, he had a IQ of 100. When we retested him, it's now 60, which then made him eligible for the IDD diagnosis. And we were devastated. It came out of the blue for us. Like, he wasn't necessarily making a lot of progress, but we weren't seeing any regression. Like, it wasn't like he lost his potty training skills or his reading ability. So it came out of left field for us that they saw regression in that aspect. So we clearly ran to our developmental pediatrician, and she said, let's revisit genetics and kind of go a step further. And then that's how we found out.
Yeah, so the workup for when you start seeing, regression of not necessarily that you were losing skills, but you had this test that told you something, and you were having some of these other features, then combined with the potential, facial features that may not be well understood or recognized by the majority of clinicians, because understandably, they're not seeing patients with these diseases. So, I think that, advocacy wise, how do we encourage the more routine testing of the various forms of Sanfilippo for children who are having learning and behavioral issues and may have some other, physical features that could be suggestive. There's certainly more use of whole exome and things like that would pick this up. And so, are you seeing a lot more children getting diagnosed by way of whole exome now, Dr. Sanchez?
I'd say yes. And that will become the case more and more. Thus far, to get whole exome, we generally have had to jump through many hoops. So we've had to do the basic CMA and many other tests in order to get the insurance to approve whole exome sequencing. That is very, very quickly changing, and it's becoming much more available, I would say, in the past just a year or two. So I suspect that will become the main way. But in the past, it has taken, you know, somebody wouldn't get whole exome for the first year or two of their diagnostic journey, and that's a long time for these kids.
Yeah.
Yeah, our insurance denied our genetic test. When we went in and saw the geneticist, I think she pretty much spotted it early on, just the facial features. She asked to see pictures of his younger days to see how his coarse features developed, which we just were like, oh, he has the greatest eyebrows and the greatest lashes, you know, and none of us have that. So we were always like, oh, the boys get the good stuff. And then other things that we never noticed was like he couldn't extend his arm. His elbow is like at a permanent kind of sub L. And she would ask us and we were like, oh, we never noticed. We kind of just worked around it. And some just like other signs and questions that they asked during the initial diagnosis. And she was like, I don't want you to look this up, but I'm thinking it's something I'm going to test for this. I'm going to put in for your insurance, but they're going to deny it. So this drug company is doing free testing for this. You know, she didn't want to tell us in advance. And I said, she goes, and I was like, I can't leave here and not know. And I promise I won't look. And of course I did. But she didn't say Sanfilippo, but she just said MPS. And so looking at MPS, there's, you know, multiple that are really devastating. So of course we waited and we found out like the day she called us in. So we knew it was something the day before, 2 days before Christmas, before we went on break. And in hindsight, I'm like, she totally could have waited. We had an appointment on the 6th. Like we had to celebrate the holiday, like around family, and each of us would get up and go to the restroom and we knew we were crying. And it was like, you know, having to absorb that and just being around a lot of people you weren't ready to tell. And my mom noticed it. My mom lives about 100 miles away. So she comes a couple times a month and she's like, why can't he do this? Why can't he do that? And so I did tell my mom because I was like, I can't take it. She's asking me like, he used to play with the iPad. He used to do this, you know, her same routine. And finally I told her and she couldn't believe it, but it just took someone. That's why these forums and this education and the people that are promoting the TikTok videos, like if TikTok and, you know, people that are active on social media, during my search 5 years ago, I probably would have made the connection because as soon as she mentioned it and I saw it, I was like, it's everything. It's literally everything. But that's why these forums, educating other physicians, is just pivotal to Sanfilippo and other rare diseases. So,
thank you. I wondered if we could just thinking how we're kind of looking towards the future of treatments and how we might think about what's important from your guys' perspective to have these treatments do. Okay. We recognize that nearly every child with Sanfilippo is being diagnosed after they are having symptoms because we yet to have newborn screening. So kind of taking into account where your child is at this point in their symptoms, you know, what are the most challenging things that are, you know, about Sanfilippo and their symptoms that are happening in daily life right now that maybe a treatment could address for you? Christi, you want to kick this one off?
I would say probably sleep. As I mentioned so many times to people, we've seen a lot of decline in Levi, but right now our main thing is sleep. He can function on 1 hour of sleep. Last night he had, not last night, the night before he had 8 hours of sleep. And I think he was just basically catching up on sleep and just still never fully is rested. So every day for us is trying to make him comfortable to where we can actually go out in public to do something where he's able to move his body and be awake. We were out last night and he was the happiest guy ever, but I'm like, you're functioning on 3 hours of sleep. And that's really difficult on our family because he's happy. Here we are walking around like zombies. And my thing is, if anything could ever happen right now would be sleep with us. That's our biggest thing, is just sleep. Like right now, I am delirious, no sleep.
Yeah.
But yeah, that's the biggest thing with us, I think, right now, is getting some rest and letting him, let his brain, his body, everything just kind of relax and rest.
Yeah. Sybil?
I would agree with the sleep 100%. So we try every means, and sometimes they work for a short period, but there's always a period of no sleep for everyone. Also, he has a lot of the gastrointestinal issues with, constipation to diarrhea. And so it's just always in the most unexpected, the worst places to have accidents. He has them. So I would say, managing that and having it not be such, it goes from one extreme to the other within a day. And you're just like, you try all the remedies, pain management, because they can't articulate it. And then just behaviorally, it's hard for us to go to church. It's hard for us to go to a restaurant or out in public. The whole time, you can't really enjoy yourself. You want him to go out and experience stuff, but the whole time we're doing like managing behaviors. So you never really fully getting to enjoy your meal. You're worried about everyone being disturbed because they're paying for the movies, they're paying for food, and he's shouting at the top of his lungs, not in anger, but just because he wants to. Throwing things, putting things in his mouth. So a lot of the things that keep you from really wanting to do stuff. I mean, even going to friends or family's houses that have like non-Solomon friendly environments where you have to just keep him seated through a whole party because you don't want him to get into anything, So it's just constantly day to day things you adapt to. I've been thinking about this because it's kind of like just commonplace. Like, I don't feel like he's up to it, and some people understand, some get upset, and I can't really manage you being disappointed in us for not coming to your child's party. It's just, he's not going to be in a space, and I already know the scenarios. Nobody's going to enjoy themselves. He's going to be a distraction. So there's a lot of moments like you want to integrate them into, but because, some of my family live 2 hours away, we have to make multiple restroom stops. He gets car sick or anxious in the car. So it's just dealing with just his discomfort level. Everything is surrounded. We always say he's got people, because two of us are, putting on a bib, and one is combing his, he's just
an entourage.
It's an entourage, all encompassing and caring for him or looking out for him. And this weekend we were with our family on vacation in San Diego, and we were at a resort, which was perfect because we were right on the beach and our room was right there. Usually if we go to the beach, we're like worried about the disgusting restrooms, so we have to plan even our vacations so that we can include him. But, it can't be too far. Everything I have to plan. Luckily, I live in California by the beaches. So everything is a vacation within 2 hours. So, and it has to be a situation where it's an even floor. He's lost some of his vision. So he has to be, he can't wander by himself or even play in an area that has any type of obstruction. So it's just day to day, even packing to go out is like it's more stuff than when he was an infant, so it's just those type of things.
Yeah. The somersaults that families have to think of, plan of, are probably, hard for a lot of people who don't kind of live life with someone who has special needs or Sanfilippo to really kind of fully conceptualize what that is. Dr. Badell-Graum, I want to pull you into the conversation and give you a chance to ask the families something from your research that you found to see if it translates into their lives.
Thank you. I actually did have a question here. Thank you all for what you were sharing. It's very interesting for me to learn about these things, and hopefully I can then bring this to the lab and try and hopefully help my research. And one question I had for maybe the parents or Dr. Sanchez maybe is, you've been mentioning the sleep issues or the gastrointestinal issues, but I wonder if you've noticed kidney issues, any issues with the kidney function or kidney in general?
We have not really seen anything like that.
No.
Ours is basically sleep and GI. GI meaning constipated one day.
Yeah.
Day is, you never know.
Thank you. And then another question I would have was medications. Apart from, are there any medications that the children are on a daily routine that they're taking? Anything like, symptomatic that they're being given, or anything that they're taking at the minute?
My son has a whole list of medications. Not really medications. We try to do mostly supplements. Medications I try to stick away with, but we do a lot of supplements.
Go ahead. Sorry.
To help with like the activation and just little things like that. Just
Mm-hmm.
We're still on the genistein from, for years. We've been doing that. I'm terrified to take him off of that. We do a very small amount, but it's just in his system still. We do just a lot of supplements for GI and things like that. Sleep, we do a lot of supplements for that. They don't work, but maybe they are a little bit. They're giving us a little bit of relief there.
We do supplements as well, but also he's on Diamox.
We are on that as well.
Yeah, because he's having, we call them episodes.
Mm-hmm.
The neurologist and sleep doctors and seizure doctors ruled out seizures. So they don't really have a name for it. So he did it overnight. He has these episodes where he has kind of like these just head drops and his eyes roll up and then he snaps out and then it's like it never happened.
Is that only at sleep time, or?
It's usually, it's when he's overexcited.
Okay.
Or just between 4 and 6, the sundown time, he's pacing and we know it's building up to it. It's almost like a release of pressure. So other parents had recommended Diamox, which we went from having like 3 or 4 hour sessions. It went from like once a week. And then and then we had this overnight with neurology and I don't know what they call them for seizure doctors. And he had them because he was having them so consistently all of a sudden out of the blue. And one of the parents on the group said, try Diamox because it's probably the buildup. And instantly it worked. I mean, like literally stopped it. Now, I don't know if he's just become accustomed to it. He has them, but it's not as frequent as that one time where we were like, oh my God, this is his constant state, 2 or 3 hours. There's nothing you can do to stop it. He's fully can converse with you. They put all the probes on and it's not seizures. So we're like, it just looks painful. And anytime it happens and someone's here, they just start crying. And I'm like, don't start crying because I'm going to cry, because it looks traumatic. But the Diamox helped. And then he's on gabapentin for, pain management. So it's just really hard to gauge if that truly helped, but at this point we've maintained it.
I think the movement challenges are something that's a huge gap in our understanding about Sanfilippo.
Mm-hmm.
And, from the focus groups and things that we've done, certainly we hear a lot that, that is a symptom, at least kind of, just taking Sanfilippo as a broad brush. If you have movement issue, disordered movement types of things, that they're very much impacting quality of life.
Yeah.
Now, I don't know if, it becomes so hard to define and get a clear answer as to what a symptom is. And, I think we as parents really struggle with it. I can imagine, Dr. Sanchez, that's really a struggle from a clinician point of view too, is kind of not being able to put it in a box.
Absolutely. We have a really hard time with not knowing answers, which we very frequently don't, especially in disorders such as this. And sometimes we end up with kind of broad approaches. Diamox, acetazolamide, this is a very old medicine that we sort of throw at everything, and it's used to treat a lot. And so a lot of times we use this medicine and it works beautifully, and we just don't know why. But we may be covering many bases. And a lot of times in rare disorders, we are following your lead. You know, y'all are on these message boards and very interactive with groups of other parents that have had experience and say, can we try this medicine? It really worked for them. And we try it because we don't have other great answers for you. And I know Marissa hasn't gotten a chance to answer the previous question yet, but I also, if y'all could just tack on, I'm interested in who follows as the primary consultant for your children. Is it a neurologist that's doing most of the prescribing? Is it a geneticist? Is it a PCP? And I'm also interested in hearing what the supplements are, if there's certain supplements that most people have ended up on.
You gotta go.
I was going to say, Christy, you seem like Levi's on a bunch of supplements, so I feel like you should go.
Well, we do a thing called Axio for most of it, or not Axio, LifeVantage. It is just a supplement that a friend of mine sells that we got him on. So that's something I do the full board of that. It helps with, I say quality of life, just as everything. What was the question? I'm kind of like just spaced out here. So what was it? What medication or who supplies it or?
Yeah, I think
okay. You know, I do like the supplements. I research and get whatever, you know, I think that's going to work for him. But one of his LifeVantage with the NRF 1 and 2 and NAD and stuff. And then like our Diamox, our neurologist, he will not provide that for him. So we had our primary doctor. He listened to me about what it would do for Levi and what it does do. So he decided, you know, what the heck, I'm going to give it to you. I gave him, you know, he kind of looked at all the other parents and kind of, I kind of printed out like what everybody else said was working.
Mm-hmm.
So he was like, you know what, you know your kid best. It's one doctor that has actually listened to me, my neurologist, our geneticist is great. She would provide that, but she's out of state. But most of our doctors won't provide that due to the fact they don't know the condition of Sanfilippo. But we've had a really good primary doctor that has just really listened to me. He actually was one of the doctors that said he's heard about Sanfilippo in the past. Yeah, so he's one doctor I have to say that will, if I ask for a drug, he's going to give it to me for Levi. So we got really thankful for that. Our neurologist, like I said, he's kind of not with us really on the same page with those kind of.
I think that's pretty common message about that one particular medicine, right? Is that you know,
there's no research for Sanfilippo kids on it and so forth. Well, of course, there's really no research for it, you know, because we're so rare. But like I said, my doctor, I printed out Facebook quotes or comments from parents and everything, and I'm like, read this. This is coming from, you know, real life people. So that was.
What's a balance between, you know, potential benefit and potential risk, and just being very mindful of, if it's a low risk medication and your child is suffering in a disease like this, you know,
that was my doctor.
Having that care team to really work with you on an individual level is so important.
UCLA has a complex care team. Oh, sorry.
Oh, go ahead.
A complex care team where they have a, you know, pediatrician that kind of manages it. Initially, our geneticist kind of coordinated and provided a list of orthopedics, you know, the cardiologist, all the different specialists to address and kind of meet all the, you know, start a relationship. And I will say that it was disheartening because there was something with everything. You know, he did an ultrasound and there was just stuff upon stuff. And it was like things that would be devastating for your, you know, otherwise healthy child. They were just like, yeah, we see that in MPS. We see that in MPS, you know? So it's like, oh yeah, there's some findings in his heart, but we see that, you know, his vision loss. Yeah, it's not surprising, but it's like, it's devastating. Like just because he has Sanfilippo doesn't mean it's any less devastating. You know, even I know he has hearing loss because I know Connor has that as well, but it's just been a struggle to get him. So we haven't even done audit because I figure what's kind of the point he won't wear a hearing aid. And it's just one more thing, you know, we know by what he articulates back, but there's literally every appointment, you know, we'll monitor it, but there's nothing we should do right now. It's almost like they don't want to try anything beyond just like immediate comfort care. So sometimes that gets disheartening because it's just kind of like discounting those things. Or maybe it's just the way that they present it, where it's just like, oh yeah, the valve is short in his heart or whatever it is. And it's like, but we see that, he has MPS. So yeah, of course he's going to have that. So it is disheartening to kind of just feel like those things are discounted, knowing that if my other kids had it, there would be a little bit more of a reaction or support in that versus, yeah, that just falls under the checkbox list of potential things with MPS, So I think for clinicians, that would be my one go-to, is kind of just some sensitivity to that. I know that we come in and we're strong because we're dealing with it day to day, but we're devastated when we leave your office, like we don't want to have a meltdown in the office, but sometimes those things like really hit deep. They're very devastating to us, and we know the diagnosis, but it's just kind of, flippant in just the approach and how it's presented to us. Like we should just accept, like, that's one of the other things.
Yeah, definitely. Feeling dismissed or written off because of the diagnosis itself and the lack of disease modifying treatments is, something I think we all feel universally. I don't think that's intended, right, Dr. Sanchez, but it is something that is quite commonly perceived by parents in those settings. It's really hard.
Absolutely. And that was part of what I was wondering about, who y'all are following with. For the most part, have y'all remained with your home institution, with the neurologist or geneticist or provider that diagnosed you, or have y'all been sent somewhere else with somebody that sees rare disorders, or how is that working for y'all?
So we actually got the diagnosis through a geneticist at DuPont. We started our journey at CHOP, but just because of our location, DuPont was actually easier to get to. So we started there. That's where they gave us the actual diagnosis, but they weren't specialized in it. So then as soon as we got the diagnosis, we did the research, realized that CHOP had a lysosomal storage specialist, and reached out to her. So we're really connected with her now. And honestly, like, it's really interesting. I'm not so much in a lot of these. I'm in the support groups. I don't follow a lot of it. And to be honest, sometimes I wonder if I don't know. I'm listening to Sybil, I'm listening to Christi, and it gets me upset because I sometimes live in my little bubble. And then I listen to you guys, and sometimes I feel really lucky, and then I feel bad that Connor is higher functioning, because I feel really fortunate. I'm listening to you guys talk about your care, and we have a really, really good team. I don't feel dismissed. In fact, the last time we went to CHOP, I think it was October, November, and my God, Mike and I walked out of there like high-fiving, like he's doing really well, So we have a really good team. We did all the baseline, same thing that Sybil said. We did, orthopedist and the cardiac stuff and the hearing, and did it all. And other than him needing hearing aids, and we have inserts for his to keep his legs, because they're his knees are bowing in a little bit. That's, knock on wood, where we are right now. But it's hard because I get on the phone and then I listen to all of you guys, and I feel a little guilty that Connor's doing as well, but then it could turn around and change tomorrow. And then I don't sometimes realize how lucky I am with the kind of care and just the team that we have. But yeah, we go to CHOP, and it's lysosomal storage department that we have.
Don't feel guilty.
Don't feel guilty at all. We're in a good place now where everything is. So, I mean, we had to fight to get where we were. So, but yeah, never feel guilty. Be honored. that Connor's doing as well as he is, because I love following Connor. He's amazing.
I'm like, no, I have to knock on wood every time. I'm like, so super suspicious.
Watching him a lot of times brings back memories for me with Levi, because we've lost a lot of those skills. So when I see Connor, it, you know, there's so much alike, but they're so different. When I see things with Connor, I'm like, oh my gosh, it brings back happy memories of what we got to cherish with Levi. So,
well, that makes me happy, because I never know, you know, I'm always like, oh God, I hope
throw him out there and let him be seen.
Yeah, he's such a joy.
Because I still wish that we had Facebook and cell phones to walk around like you guys do with, you know, we didn't get that with Levi. We had, you know, the camcorder when he was younger, you know, so we had like TV station following.
Right.
Take advantage and just show him off, because I wish I could have access like on my phone and computer of all those memories. So like I said, I love watching everybody's kids grow up. It's amazing.
Thank you.
Yeah. I just put a plug into Dr. Sanchez's question that global consensus clinical guidelines are being developed, and there'll be a presentation later today or tomorrow on that. So hopefully that will help provide additional support to any clinician anywhere in the world to have access to, you know, at least a starting point on direction of care. I wanted to circle back to something that we had talked about in kind of our previous conversations with the parents, and some things came up as far as quality of life symptoms around vision and mobility. And I think that would be really interesting for everybody to hear about that type of symptom in Sanfilippo type C. Christi, I know you had some comments on that.
With hearing, Levi has mild to moderate hearing loss. I don't see it.
With vision.
Oh, with vision. Okay. Yeah.
Vision,
Like I said, I'm like running on like.
The audio's a little funny. Sorry.
Eyesight. Yeah, I did not write down the name. I think you remember the name, Cara, that Levi was diagnosed with, the pigment.
You had told me that he'd been diagnosed with retinitis pigmentosa.
Yes. Yep.
So
with that, and we don't treat that at all, because that's, you know, there's really nothing we can do with Levi. He's not going to wear glasses. He would just take them and throw them. So, but with his eyesight, we've noticed a lot of times, like at nighttime, when it starts to get a little bit dark, you know, in the evening, he cannot see, cannot walk anymore. And we thought it was walking and the neurological, but it's really like when the lights go down, he cannot see at all. It's like blinders go on. So at nighttime when he gets out of bed, the fortunate thing is he don't leave his room. He stands there, because it's like walking in a black room. He can see well, like we play test, you know, or move this, but he really does have the eye issue, or the sight. Walking up over steps, we have to remind him like we're coming up on a step, so he can get his leg up, or like when the carpet changes. If it's like squares or, you know, just any kind of like funky design, he literally will stop and has to really think hard. Left foot, right foot. He can get through it, but sometimes usually what we're doing is my husband and I are both holding on to him, and like going into a movie theater, he looks like he's like this hunched over kid and he can't see. So we're dragging him in through blackness. He has a really difficult time with his eyes. But if it's daytime, he can see as well as anything until the carpet or the sidewalk.
You have a transition or contrast.
Yeah. Transition is.
Well, how about you?
Solomon's the same, and his he saw a genetic ophthalmologist actually who knew they existed, but he said that seeing is like this for him. He could just see
right.
Like,
no
peripheral. And so everything's out of sight, out of mind. You know, it had to change. It changed everything we have to do. It has to be like right up near his face.
Yeah.
Even eating food, he's scooping like an empty spoon because all the food moved over, you know. And the same thing with the shadows and the steps. Like you have to help him and tell him step, because he can't see them. Right. And we went on vacation and it had an upstairs, downstairs, and we didn't realize the magnitude because he's used to our steps, right? He does them every day. He knows exactly. but there was
He
pattern on the
almost like a blind person. They
yeah, he got to the top and he was still doing the like step walks, and we were like, what's going on? That's it. Like, why are you still stepping up? So it was kind of shocking because, like you said, he can see fine forward, but the same thing, his alarm, he won't leave the room. He'll just stand by the door and be like, boop, 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 boop with the light switch. Yeah. Where before it was 2 and 3 in the morning and he's like running in circles. Now, I think because of the dark, well, I know because of the dark, he won't go beyond his room, which is helpful, but we always get the sign because he starts flicking the light like our alarm clock, come get me. But yeah.
Yeah, no, I think that's really interesting, and maybe something as you kind of take that information back to the lab, you know, kind of correlating that with some of the findings. Have you looked at the eyes, Dr. Badell-Graum?
We actually haven't looked at the eyes at all, but it's very interesting to know because it's something that is seen in other lysosomal storage diseases. So yeah, I've actually made a note of it. It's very interesting to learn about.
Jill, I believe, did a study.
Yeah, Jill did something on the eyes.
She could probably send to you.
Yes, there certainly has been some work done around that.
Patients with mutations in the same enzyme that just have retinitis pigmentosa without Sanfilippo. So that is part of what has sparked some of that interest. And what you described is very classic for retinitis pigmentosa, where you tend to lose your night vision first, and then it's your peripheral vision. And it presents exactly how you're saying, where in the dark you have more mobility issues, and then things like stairs and things that require your peripheral vision become an issue. That on top of the other mobility issues could severely limit mobility in these kids.
Yeah, because at first we almost thought it was mobility or something like that because his legs weren't going, and and then we really realized that, I mean, there's mobility kind of in some parts, but it's mostly the eyes that affect his mobility, I think.
Yeah.
So he's always got to have 2 people when we're in different places or something, just to kind of be able to get on walking around so he does not fall and hurt himself.
It's definitely something I think I'm going to look into here in our work, because we have all the setup to look into different eye things for other diseases that we work on.
For a Sanfilippo kid or a nightmare. We went and seen every eye doctor, and yeah,
that's a problem.
Yeah, so I think it's interesting. Sorry, go ahead.
That's a problem that we've been encountering, that one of the main tests that we would want to do for this issue is OCT, which is a test that is very technical and requires some cooperation. And we're thinking for this population, we might not be able to do one of the main tests that we would like to do to look for this.
What is your experience or availability? I understand that there are now some, handheld things, and, is that something that can be done under anesthesia if a child is already, has to go under anesthesia for something, and this is a fairly quick test. Is that correct?
You might be talking about OCT. That is, I believe, a handheld thing, and I'm not an ophthalmologist, but my impression is it's technically pretty difficult. It's not a long test, but it's technically difficult and requires a person to be able to hold still and do what's needed. And we did talk to our institution about, is that something that can be done under anesthesia? And it sounded like the answer is no. I don't want to say that's going to be the case everywhere, but it sounded like the answer is no, because we wouldn't get a good test, not because the institution won't do it or sedate for it.
Oh, yeah, you should have seen us trying. You should have seen the crew that was like trying to get, we got like one good image the first year on one eye, and then luckily the other one, but all the rest was like, and hollering, and it was quite the show.
You come out sweating like you've been at the gym for an hour.
Oh yeah. My husband are like fighting.
But I think that just challenges us to either think about other ways that we can navigate getting an important test done that may lead us to understand clinical outcomes of future treatments, Rather than just saying, oh, we can't do that, so we're not going to, assess that organ. We have to really start getting creative about how we can, understand that disease impact in our patients over time. So I think that's a challenge to all of us moving forward. This has been such a wonderful discussion, and I thank all of our panelists for participating, and in particular our parents for sharing such personal information in this public setting. I know you have really informed so many people, and they'll take a lot from what you shared. We have to wrap up now and move on to our straight into our lunch and learn session. But thank you so much.
Bye, thank you.
Thank you so much.
Thank you. Okay, let me introduce our next session about advancing newborn screening. And I'm so thankful to have with us today. First, we'll have Dr. Matthew Ellenwood, Chief Scientific Officer at the National MPS Society, followed by Dr. Melissa Wasserstein, Professor of Pediatrics and Genetic Medicine at the Albert Einstein School of Medicine. Dr. Ellenwood, take it away.
Great. Thank you so much for this opportunity to come speak to you about newborn screening, which is such a critical linchpin for helping us advance future therapies. Could I have the next slide, please? So as a very basic overview, I'm going to be in a somewhat rapid fire way, going to be overviewing basic principles governing newborn screening in this country, the entities that oversee and regulate and actually conduct newborn screenings, how conditions are selected for newborn screening, and where we are in this process for Sanfilippo syndrome. Next slide, please. So newborn screening is conducted with great mindfulness of considerations for ethical, legal, and social implications. And that's because it is not simply a test of a newborn. It is a test that is done at a state level in the United States, and it is a public health measure. So all children born in a given state will be tested. And this requires a certain bar be met in terms of how accurate and reliable a test is, its ability to be used on a population level, so it has to be scalable, and you have to have the ability to both confirm a diagnosis with a minimum of false positives, so you're not causing anxiety and worry to families for no reason. There also needs to be a very clear necessity for early diagnosis and urgency of treatment in this case. And there needs to be an effective treatment. And that could be an accepted treatment, for example, bone marrow transplantation for MPS I, or an accepted and approved drug, for example, hyaluronidase for MPS I would fit those needs. Next slide, please. Where we are for Sanfilippo. So in terms of actually conducting the tests, these are usually done by an analytic technique called tandem mass spectroscopy. And we have all the reagents that have been developed at the laboratory level for MPS IIIA, IIIB, IIIC, and IIID. These have all been published on. The substrates are available commercially for MPS IIIA and IIIB. IIIB is available through Perkin-Elmer. MPS IIIA substrates are available through a private company founded by Michael Gelb, who's actually created most of these substrates. Unfortunately, we do not have any treatment known for any of the Sanfilippos that effectively address the etiological causes, and no drugs are available to do so. There are hopefully many clinical trials ongoing, not many, some. Gene therapy, in particular for IIIA, and CNS administration of ERT for MPS IIIB are currently in trial and extension phase. And there are promising approaches to treat brain disease by targeted ERT in the pipeline for MPS IIIs. Next slide, please. So testing. who decides and how do they decide whether to start testing? So mandated newborn screening is determined and conducted at the state level in this country. We have a federated program. So the states are the ones that are authorized, and they make their decisions individually. They have their own method for determining what to screen for. Some states do have what we call a mandate based on advocacy lobbying. So for example, advocacy lobbying got MPS 1 as a mandated test in Illinois. So there is a place in some cases for direct lobbying of legislatures. There is a federally recommended newborn screening list. It's overseen by the Department of Health and Human Services, and it has input from the Advisory Committee on Heritable Disorders in Newborns and Children. And you may have seen that acronym ACHDNC. The ACHDNC oversees what's called the Recommended Uniform Screening Panel. You may have also heard that referred to as the RUSP. And they oversee changes or additions to this recommended panel, and there is a defined and detailed nomination process. These recommended nominations must be accepted by the Secretary of Health and Human Services. So the ACHDNC doesn't have the last word. It has to go up to the Secretary. The RUSP, again, is only advisory to states. States don't have to listen to it, although some states do have what we call alignment legislation. Next slide, please. Is a state-based mandated test the only approach? No. There are what we call research approaches that can be done, and Dr. Wasserstein will cover one of those. These are so-called pilot studies. Note that for the determinations of a nomination, pilot has a very specific meaning. These require ethical oversight and approval with informed consent. They may involve identified samples of patients. Some of them may be de-identified, just working out a laboratory protocol. Usually these are based on a regional or a hospital or a network-based group of hospitals in terms of their scope. Next slide, please. So how does a condition gain RUSP nomination? A nomination will require the following to be successfully submitted. You need to have a reliable, sensitive, and scalable screening test. There needs to be a consensus on what is a confirmatory test. There needs to be medical urgency in the newborn period, and there needs to be an approved or accepted and available treatment. And there needs to have been a prospective population-based pilot study that fulfills the one patient rule. There needs to have been one patient identified in a screening pilot of a population that is prospective, population-based, and that patient needs to have been confirmed and been treated successfully. There are other additional details of diagnosis and confirmation and practice that need to be in place, but those are the big ones that are required to get a nomination started. Next slide, please. Once a RUSP nomination is submitted, it goes to HRSA, which is an agency of HHS. They will confirm that it is complete and then forward it to the chair of the ACHDNC. The ACHDNC will then assign it to what's called the prioritization work group. They review the nomination and see if it's ready to go to what's called evidence review. And that usually takes a minimum of 3 months between meetings of the ACHDNC, which occur 4 times a year. Once the prioritization group presents it to the entire ACHDNC and they vote to move the nomination forward, it has 9 months to be reviewed for its evidence and for the ACHDNC to vote on it. Once they vote to recommend that the condition is added to the RUSP, then it goes to the Health and Human Services Secretary for signature, and there are 4 months that they have to sign it or provide reasons why they cannot accept it. And that is sort of the basic outline of the process. Next slide, please. So even if you get RUSP nominations approved, it's not the end of the road. As I said, the RUSP is not a mandate. Each state has to decide whether they're going to start testing. Some states have what we call RUSP alignment. In some states, RUSP alignment means it's nearly automatic that within 2 years they'll start testing. Other states, that RUSP alignment may be a little less clear, and there may or may not be a sure path, and it may take up to 3 years for that addition to be made, and there are other possibilities that could delay it. To give you an example of how these things can work, NPS 1 was approved for addition to the RUSP in February of 2016. Currently, and projecting 3 years hence, based on states that have RUSP alignment legislation, we'll only be seeing about 75% of the population of children in this country who have the ability to be diagnosed in the newborn period for NPS 1. So even if we get a RUSP nomination approved, it's not a sure thing that it will be available to every child born in the United States, although it's one of the best approaches we can take to ensure that that happens. Next slide, please. So the path forward for Sanfilippo syndrome. There are 2 elements currently limiting the submission of a nomination for Sanfilippo, and that is an effective and or an approved therapy for any of these disorders. And that will sink any nomination we were to try to put forward. There needs to be an effective and an approved or accepted therapy for the disease in question. The other is we have not fulfilled the 1 patient rule at this point. And this is where the next speaker is going to be so important because Screen Plus is actually incorporating MPS 3B into their panel. So as I said, the other issue is we need to find a patient who has been identified in a screening protocol. They need to have been effectively documented, confirmed, and treated. So those are the 2 big hurdles that confront us. And what should we hope for as an optimal nomination schedule? In my mind, it is that we have everything, all the ducks in a row, so to speak. So the minute that that patient comes through and it gets treated, and the minute that that treatment is accepted or documented or approved, we get this nomination in as quickly as possible. That would be the most obvious and beneficial approach. And I think that that concludes my slides. To the degree the slides are shared, next slide, please. There are a number of links if you would like to learn more about this whole process. Thank you so much for being able to overview newborn screening for the NPS disorders.
Hi, everybody. It is a pleasure to be here to present Screen Plus, and I really want to thank Dr. Ellenwood for that really perfect introduction to Screen Plus. As he mentioned, Screen Plus is a research program. It is a pilot newborn screen, which means that we're evaluating how well newborn screening works for certain disorders, including MPS 3B. Next slide, please. So here are my disclosures. As I'll show you, Screen Plus is a multi-sponsored program, and this slide shows our industry and patient advocacy group sponsors. Next slide, please. So I'm gonna, in the few minutes that I have, gonna give you a big kind of bird's eye overview of Screen Plus. I'll introduce you to our informed consent and recruitment processes. Again, it's a pilot study, so we do have informed consent. And then I'll also share with you our financial infrastructure, which is one of the unique aspects of Screen Plus. Next slide. So Screen Plus is the largest consented pilot newborn screening program. We're running it in conjunction with the New York State Newborn Screening Program. And our goal is to enroll 175,000 babies born at 9 high birth rate ethnically diverse hospitals that are shown on the map here over a 5-year period. We want to see how well newborn screening works for it, how well the assays work, and we're also evaluating the ethical, legal, and social issues associated with newborn screening for complex disorders. Next slide, please. This slide shows our initial panel for Screen Plus on the right. Our panel is fluid, which means that if a disorder is added to the RUSP and also instituted in New York State during the course of Screen Plus. We can remove it, and we can also add new disorders if they meet our criteria, and we have very specific criteria for disorders to be on the Screen Plus panel, and those are shown here, and those include that we have to have an assay that works, that's been validated, we have to have a disease that has significant morbidity or mortality, it has to have a pediatric phenotype, and importantly, it has to have either an FDA-approved treatment or treatments that are currently in clinical trial. Next slide, please. And as I mentioned, we have a big goal of evaluating some of the ethical, legal, and social implications. And this starts right after birth. When parents are approached by our study coordinators to participate or not, we have a consent feedback survey where we're asking people how well we did, how well they understood the program, reasons for participating, reasons for declining, and we're collecting socio-demographic factors. We have a series of quantitative parent surveys which are being sent to participating parents about 1 month after they get the baby's results are returned. And each survey has a different topic, and we have a bunch of different surveys, and they're asking about how do you feel about expanded newborn screening? How do you feel about screening babies for later onset conditions, for untreatable conditions? How do you feel about using dried blood spots, that are the residual dried blood spots that are stored at the newborn screening programs? How do you feel about sequencing babies' genomes at birth as a form of newborn screening? And I think this is one of the strengths of the program, is that we're going to have access, as I mentioned, 175,000 families. Even if a fraction of them answer these surveys, it's still going to be a massive amount of really interesting data that will hopefully help us expand newborn screening in an ethically appropriate and acceptable way. The third part of our LC studies are qualitative interviews, and we're really focusing on parents whose babies have either had a positive or uncertain result. And the reason why we're doing this is because in my own experience as a newborn screener, we always focus so much on the babies and making sure that the children are plugged in and getting the care that they need, that sometimes I feel like we don't really give the parents the support that they need. So our goal here is to better understand what parents are going through so that we can then share that information and offer better support as a whole. Next slide, please. Next slide, please. So our informed consent for Screen Plus. We have a full-time coordinator at each of our 9 pilot hospitals. Our method is that each coordinator has direct conversations with parents, which last approximately 15 minutes. Most of our coordinators are bilingual, speaking the 2 most common languages at the hospital. Usually English and Spanish is the most common one here in the Bronx. Our brochures are shown here on the right. This is 3 of the languages that we're using. We also have a novel e-consent. We have IRB-approved scripts and these brochures. All of our study materials are currently translated into the 8 most commonly spoken languages at our pilot hospitals. If there's somebody who doesn't speak one of those languages, we have translation services available. And then again, we're getting feedback on how this is working in case we need to modify it to improve it. And we're really carefully tracking how well we're doing with recruitment. Next slide, please. So we launched back in May 2021, and we did in person, where we had our recruiter knock on the door of a family who had just had their baby within the first 24 hours and discuss the study, introduce the brochures, study material, potential benefits, risks, limitations of participation. And we had overall a 64% consent rate. We then had a little transition where we had to switch coordinators. Our coordinator went off to medical school, so we had to hire somebody new. So we had a little lapse for a few months in the summer. And then, as you'll see, we had different recruitment models during launch 2. And this was because COVID hit, and COVID hit the Bronx terribly around Christmas time in 2021. So we had to pivot on a dime to switch recruitment methods where it was no longer safe to go into the room. So we switched to hybrid methods where we would call, leave brochures for the families, and then we switched back when the numbers went down. So we kept flipping different ways of recruitment. But the point of showing this slide is that despite this overall 800% increase in COVID positivity during these transitions, we actually had a fairly stable consent model. I think our ability to do these transitions were thoughtful and they worked. So the overall consent rate was actually still in the 60s, which was very reassuring that we weren't taking such a hit from COVID. Next slide, please. And as I mentioned, we do ask parental feedback about how we're doing with recruitments. This is just some early data. Most of the parents said that their reasons for participating was that they wanted to learn about any conditions that their baby might have. The second most common reason was more altruistic, that they thought that it might be important to help other families by participating in this research, which might have enhanced newborn screening. We asked them what was the most helpful material that we gave to help them make the decision to participate, and 72% said that it was these one-on-one discussions and the opportunity to ask questions with our recruiting team. We asked why they chose not to participate, and we only had a few people who answered this part of the brochure, the survey. But the main reason was actually concerns about their baby's privacy, which made us kind of readdress how we're presenting privacy issues and data security with them. Our demographics are shown on the right and generally are similar between the consenter and decliner group and generally represent the population in the Bronx. So we are targeting the population that we're trying to get to. Next slide, please. And then are just a couple of slides about our financial infrastructure, because it is one of the fun things about Screen Plus. We knew that something like this, with all of the personnel costs for our recruiters, was going to be incredibly expensive. So we submitted a grant to the NIH to fund the study, but we also realized that we needed to engage other stakeholders to help support the program. So we now have a total of 15 entities sponsoring the project. The industry sponsors and advocacy group sponsors are largely sponsoring the patient recruitment efforts and patient follow-up efforts. NIH is largely sponsoring our coordinating center and our LC studies. We had very careful stipulations for our industry sponsors that basically keep it an independent, scientifically clean project. So they're not getting anything identifiable, they're not having any role in the study design, and importantly, any therapeutic decisions for any positive child would be made independently with no obligation to use sponsored products. Next slide. I'd just like to show this because it's taken a much longer time than we had anticipated to get all of these contracts up and running. And it did delay the start, that plus COVID, but everything's pretty much done now. So it's we had a little party once we got the final contracts done. Next slide, please. So in conclusion, large-scale newborn screening projects are needed to assess the appropriateness of candidate disorders for routine screening. Most parents are eager to participate. Dynamic informed consent models can thrive despite external stressors, and Screen Plus provides an example of successful, mutually beneficial financial partnerships between academia, government funding, industry, and patient advocacy groups. Next slide. Here is my fabulous team, and there's a minute left, so I'm actually going to just put up the next slide, which is our contact information if you have any questions for me. I think we have a minute left.
Thank you both for the really excellent and informative talks. I know the Sanfilippo Foundation has been very proud to be able to help support the Screen Plus program, and we're excited to see how that's progressing and all of the other work you're doing around screening to support the Rust nomination, as Dr. Ellenwood nicely outlined for us. There's so much collaborative work yet to do to achieve universal newborn screening for Sanfilippo, and we are certainly looking forward to continuing those efforts with you both and other partners in the community going forward. We have just a moment or two if anybody has any questions. Here's your chance. Dr. Wasserstein, we have, I understand that Sanfilippo type A is certainly on the, the next tier to be added into that with Dr. Geld's work in establishing that part of the assay. Could you just fill us in a little bit on where that stands and where we're looking?
Yeah, so he has an assay that seems to be working well. I think he's right now looking for a couple more true positive dried blood spots just to help out with validation, which is something that we have been trying to get for every disorder. We can't always find them because they're like gold when we do find them, which is the so points to the benefit of keeping those residual dried blood spots. But we will be adding things in a chunk. So we have a few different disorders that are likely to be added at one point, which means that we have to, redo all of our consent materials and our programmatic brochures, et cetera. It's not too complicated, but we're waiting for all of our hospitals to get onboarded and get things running before we then have to switch everything around. And we have a bunch of hospitals that will be starting in the next couple of months, which is fantastic. So.
Certainly. And then for the other two Sanfilippo subtypes, C and D, how close are we to being able to kind of look at adding those as another tier?
We've been focusing on A because that's in the next chunk. I can have more information for you about C and D. I know he's been thinking about it, but I'm not sure where Dr. Gelb is with the assays for those two disorders.
Thank you. Okay. Well, we have a few minutes that we actually might get to take a stretch break, everybody. It's been go, go, go. Thank you so much for joining us. And we'll take just a brief pause and come back at 12:30 for our next session.
Great. Thank you for having us.
Thank you, Dr. Elwood.